far afield, which might seem far afield for biologists, but in fact should be all of the same piece, because part of this is what we call generalized Darwinism, the expansion of Darwinian thinking beyond genetic evolution to include all variation, selection, and replication processes. That's part of what this is about. And the objective here is to restore the simplicity and foundational nature of multi-level selection for explaining all forms of prosociality defined as any trait oriented towards the welfare of others or one's group as a whole. And so here's the simple idea. First of all, natural selection is based on relative fitness. It doesn't matter how well an organism survives or reproduces in absolute terms, only relative to others in its vicinity. And based on that, prosocial traits have an inherent disadvantage. They're the very opposite of competition at the individual level, which favors those that survive and reproduce compared to others. And so we have an inherent disadvantage of prosocial traits at the most local scale. But thankfully, if we just go up in scale, we can see that groups whose members behave prosocially towards each other have a robust advantage compared to groups whose members cannot cohere. Everyone should be able to understand this and appreciate this dynamic. And so, as I put it with the other Wilson, Edward O. Wilson, in a 2007 review article, selfishness beats altruism within groups, altruistic groups beat selfish groups, everything else is commentary. That's how simple and foundational it is. Now, historically, it was framed in terms of two-level selection, but it can be expanded. We can frame shift downward, and we can study competition among genes within individuals. Then, of course, we have familiar within-group selection between individuals within groups, between groups in a metapopulation. There's your two-level selection, but we could frame shift upwards in a variety of ways to, for example, groups of groups in single species, multi-species communities, and then at the more macro scale, selection operating at the level of species. And so the general rule to keep in mind is that adaptation at any level requires a process of selection at that level and tends to be undermined by selection at lower levels. That is the basic dynamic of multi-level selection. So here's the complex history. Darwin really got it right, and Elliott, I think, will be reviewing Darwin's thought about group selection. He clearly perceived that his own theory could not explain prosocial behaviors unless he added a layer of natural selection. There was a period in which not everyone was so discerning, and so they assumed the traits evolved for the good of the group, either without requiring special conditions or else assuming that group level selection was powerful enough to overcome within group selection in most cases. And then in the mid 20th century, with figures such as George C. Williams and John Maynard Smith and Richard Dawkins, that led to a near total rejection of group selection in the mid 20th century, then a revival and a rethinking of the relationship between multi-level selection and other theories of social evolution, such as kin selection, reciprocity, game theory, and so on. And so today, when you look at the biological literature reflecting this complex history, it is very, very conflicted. You can scarcely know what to think reading the biological literature, and textbooks are sadly out of date. And so Prosocial World, my nonprofit organization, this year initiated a multi-level selection initiative in order to catalyze study in this area. We now have over 60 members, and you're welcome to join us. And so if you want to join us, email hello@prosocial.world. That should be pretty easy to remember. And I feel that we're already having a catalytic effect, including this, this special event. And so if we look at the explanatory scope, the field of applications for multi-level selection, first of all, there's a macro-evolutionary component, which Mitch is going to be covering, and Joan also in their talks, such things as major evolutionary transitions, the evolution of sexual reproduction and evolvability. And then there's a large number of micro applications, cancer and pathogen evolution. Athena Actopus is going to be talking about that. We have animal behavior, ecological communities. Microbiomes are a fascinating unit of selection to think about because they're so large. Thousands of species, numbering in the billions, somehow perhaps being units of selection. Mike Wade is here to talk about animal and plant breeding, and then the adaptive management of natural systems. And so point here is that there's practical applications in addition to basic science applications. And then with humans, we also have a macro and micro level. We need multi-level selection to explain everything from human origins to the our basic capacity for culture, the increase in scale of cooperative human society over human history, nature of religion, and so on. And then microevolutionary applications, which really get into the practical realm. And I think that, as I'll be saying in my talk, one of the most important reasons to establish the foundational nature of multi-level selection is because it provides such essential knowledge for virtually all positive change efforts. The whole world needs to know about the basic dynamics of multi-level selection theory. And so here's our menu for today, and I am within 2 minutes, so that's great. Elliott Sober is next up. And I think this diversity of topics all by itself illustrates the foundational nature of multi-level selection theory. A theory that actually can be applied to all of these topics is a very general theory indeed. And so with that, I will, we're actually just at time, 10:59. And so I maybe have time to answer just one quick question, or perhaps best to proceed directly to Elliott. And Elliott, if you could introduce yourself briefly, our speakers will be introducing themselves.
Be happy to do that. Let me tell you that I've been interested in group selection almost as long as David, not quite, maybe 5 years, fewer years. I'm a philosopher of science. I'm not a biologist, but I've been fascinated by philosophical issues that arise in evolutionary biology. This is one of them. Phylogenetic inference is another one that's interested me also. So shall I go ahead with my talk now?
Yep, go ahead.
Okay.
Please.
Okay, here is the list of topics that I'll oh, let me do it. Some terminology, etc., Darwin's views, a little bit about the modern synthesis, George C. Williams's 66 book, and finally ideas from Richard Lewontin. So here's the terminology. Individual selection means selection within groups where selection favors traits that are better for the individual over traits that are worse. Group selection means selection among groups where selection favors traits that are better for the group over traits that are worse. And of course, better and worse means fitness differences. And historically, and up until the present day, the standard example, perhaps too obsessively considered, but it's the standard example anyhow, is altruism. Here's a figure I like to use to illustrate the essential facts about altruism and selfishness as they're understood in evolutionary biology. In any group, no matter how common or rare selfishness and altruism are, the selfish individuals are fitter than the altruists. But the more altruists there are in the group, the better off everybody is. And the more selfish individuals are, the worse off they are. And that thing about group welfare, group productivity, is illustrated by that red line, and that's W bar, representing the average fitness of the individuals in a group. I want to connect this biological idea with an idea that's much older in statistics called Simpson's paradox. And to explain how it applies, I want you to consider the two groups that you see here, G1 and G2. G1 is mostly altruistic. G2 is mostly selfish. Within each of those groups, the selfish individuals are fitter than the altruists, as I said. But now consider the average fitness of altruism in this two-group metapopulation. Most of the altruists are living in G1, so this red thing here is the average fitness of altruism in the metapopulation, and most of the selfish individuals live in G2, so the average fitness of selfishness is this other red dot. And as you see that, and here's the paradox, although altruism is less fit than selfishness within each group, when you average across groups, altruism is fitter than selfishness. And this is a paradox. That's the traditional word for it, because it seems crazy that it's a mistake that what's true in each group doesn't translate automatically to the metapopulation, but there's no such automaticity at all. And once you see this fact about Simpson's paradox, it's clear, it's easy to visualize why there is a fallacy in the following reasoning. Since altruism and selfishness are traits of organisms, and altruism has a higher average fitness than selfishness in this example, then altruism will evolve because of individual, not group selection. And the thing that's fallacious about this is that A is on average fitter than selfishness here, and A and S are traits of individuals. But in fact, what will happen, at least in the short term in this two-group example, is that altruism will increase in frequency, but group selection and individual selection are both at work here, and individual selection is selection within groups. It's against altruism. It's the group selection part that makes that go forward. So you can't conclude that group selection is excluded here just because A is on average fitter than S. Now some history. Darwin. Let's start with Darwin. Helena Cronin and Michael Roos are philosophers and historians of biology. Richard Dawkins, you all know who he is. And they've all asserted quite energetically that Darwin shunned the idea of group selection. He was a devotee of the individual level orthodoxy. He believed in the selfish organism. He believed that selection always worked. That selection always works at the level of the individual rather than the group. This is, I'm afraid I have to say, very poor history. Here are 4 examples from Darwin in which he introduces and applies the idea of group selection in much the way I was describing it before, without Simpson's paradox and stuff like that. The first 3 involve an altruistic trait undergoing evolution by group selection. The 4th is different. That's the barb stinger of a honeybee. When a bee stings an intruder to the nest, it pulls away, eviscerates itself, and dies. The barb keeps the stinger in place in the intruder, and it keeps pumping venom. The stinger, Darwin says, is not good for the individual honeybee, but it benefits the hive, the nest. So it's nests that have barb stinger honeybees in it doing better than nests that do not, that causes this trait to evolve and be maintained in the honeybees. Darwin's example about human warfare is possibly the most familiar example. He talks about warfare. He says, he who was ready to sacrifice his life, as many a savage has been, rather than betray his comrades, would often leave no offspring to inherit his noble nature. The bravest men who were always willing to come to the front in war and who freely risked their lives for others would on average perish in larger numbers than other men. So how did the norm of self-sacrifice exhibited by soldiers in warfare evolve? Well, tribes that had altruistic individuals in their army did better in their competition with other tribes who had only selfish individuals. So it was group selection in human history that's responsible for this norm that seems to be present in pretty much all societies, that depending upon the circumstances, individuals are prepared to give up their own welfare, in this extreme case, their own lives, or risk their own lives for the good of the group. The third example is somewhat familiar, but in Darwin's big picture, it's played a very special role. It really stumped him how he could account for the worker sterility in the social insects. And the stumping happened in the 1850s. He was quite far advanced in his development of the theory, and he hit a wall which really confounded him for a year or more. This puzzle is simple. How can sterility evolve by natural selection if individuals with the trait cannot pass it along to their offspring? To illustrate Darwin's solution to this puzzle, I'd like you to consider two strategies that a fertilized female could use. The all-F strategy is to have 10 fertile offspring, and the mixer strategy is to have 9 fertile and 1 sterile offspring, where sterile individuals help other individuals in the nest. And to keep it simple, let's imagine that nests are founded by pairs of fertilized females. So there are really 3 kinds of nests. Both founders are mixers, both founders are all F. And some nests are made of 1 mixer plus 1 all F. Within nests, there's no individual selection in 2 of the types of nests because there's no variation in traits there. But in a 1+1 nest, there is variation. And in that nest, mixer's sterile offspring helps its siblings and also helps the offspring of the all F female. So mixer is an altruist here. And when you compare, that's the within group story, selection against mixers. Between nests differ in their productivity, and the more mixers there are who found a nest, 1 is better than 0 and 2 is better than 1, the more productive the nest is. So group selection is the Darwin solution for how the existence of sterile offspring evolved. The least familiar of all the examples in my list is an example from The Descent of Man about technological innovation. Darwin says, now, if some one man in a tribe more sagacious than the others invents a new snare or weapon or other means of attack or defense, the plainest self-interest would prompt the other members to imitate him, and all would thus profit. If the new invention were an important one, the tribe would increase in number, spread, and supplant other tribes. And I think Darwin was thinking about this in the context of what we now call cultural group selection. Being a technological innovator does not occur because you have a gene for that. It occurs by teaching and learning, and that becoming a tradition that's passed along culturally. And an interesting feature of this example is that you have group and individual selection both acting in the same direction. There's no conflict between them because the trait is not an altruistic trait of being an innovator. There is individual selection for being an innovator, and there's group selection for having groups composed of innovators. Okay. Next historical snapshot is just very brief picture of how the modern synthesis really had 2 subcommunities. Field naturalists following Darwin thought that group selection was an important kind of natural selection. They didn't think it was They didn't think it's the only kind of selection that matters, but they helped themselves to this idea, for example, to explain why populations restrain their exploitation of the food supply, because there's selection against groups that don't do that. They go extinct. So it was just a part of the toolkit for these field naturalists. The situation among mathematical theoreticians was really very, very different. Fisher and Haldane didn't have much time, to say the least, for the idea of group selection. It was Sewell Wright among this trio of founding fathers of population genetics in this period who did have time for it. In this 1931 paper, Wright presents the theory of interdemic selection. So here we have a selection process in which X is fitter than Y when the frequency of X in a group is to the right of that little red line. X will go to 100% for starting frequencies to the right of the red line. And trait Y will go to fixation if the group is to the left of that. So if you imagine one big population here, it'll either go to 100% X or 100% Y by individual selection, depending upon its starting frequency. And what Wright was interested in was to not talk about one big population, but a population made of many small populations. And some of them will have starting frequencies to the right of the red line. Others will have starting frequencies to the left. The first group will go to 100%, bunch of groups will go to 100% X. The second group of groups will go to 100% Y. But then the X groups, being fitter on average than the Y groups, will found more colonies and will outcompete the Y group. So you have a two-stage process where within group selection, individual selection results in monomorphic demes, subgroups, and then groups competing with groups further changes the composition of the metapopulation. And for Wright, this was a key concept for understanding how groups can escape from a local adaptive peak that's not the optimal adaptive peak. In this interdemic selection case, in principle, the whole metapopulation could go to 100% X. Wright later on tried to apply this idea to altruism and selfishness, and I think he realized that this did not work out so well. Individual selection is not going to lead any group to become 100% altruistic. That would happen only if there was random genetic drift in small populations evolving against the tide of individual selection. For that to happen, these groups have to be isolated against migrants. There has to be very little migration. But then the question arises, if a deme becomes somehow 100% altruistic, how's it going to export its altruism to other groups? And won't it all get swamped? And I think this idea did not work out. And I think Wright realized that this was not an adequate solution, though he was convinced that group selection is needed to explain the evolution of altruism. Now I turn to the bombshell of the 1960s. As David mentioned, adaptation and natural selection was a giant argument against group selection. Williams was not the only one, but he's the guy I'm going to concentrate on in my talk. His book has got a mixture of different kinds of arguments against group selection. Some of them are a priori. They bring up very general considerations that show that evolution by group selection is going to be very rare. And then there's an empirical argument that he offers, which is about a specific trait, and he uses that to try to evaluate whether group selection is the right answer for at least that trait and possibly others. So here's the first example of what I mean by an a priori argument. I call it the representation argument. It says evolution is change in gene frequencies, so evolution by natural selection must involve genes differing in fitness. Therefore, the units of selection is the meiotically dissociated gene, not the organism or the group. So just by the very definition of evolution as change in gene frequency, you get this result. The argument is flawed, I think, because the fact that a gene evolves to fixation leaves open whether it evolved because of group selection or individual selection or some other kind of selection. And nothing follows from the fact that evolution, if it is a fact, that evolution is changing gene frequencies, that tells you that it's genic selection, not organismic or group. A more interesting argument, I think, in Williams's book is based on parsimony, the principle of parsimony, Occam's razor. And he has this hierarchy in mind here that neutral evolution is more parsimonious than individual selection alone, which in turn is more parsimonious than individual plus group selection. I think this parsimony ordering is correct, but it's incomplete because it doesn't include a consideration of where group selection only fits into it. And what you see here is, I think, a fuller representation of the parsimony ordering. The simplest model is strict neutrality, zero variation in fitness. That's a null hypothesis in the literal sense of null, zero difference in fitness. Individual selection will involve one kind of variation within group variation, group selection between group variation, and then you have the most complex model that brings individual and group selection together both. In the part of statistics called model selection theory, the complexity of a model measured by the number of adjustable parameters it contains is relevant to evaluating how good the model is, but so is fit to data. And with this, I think, better picture of the parsimony concept in connection with units of selection, I think we can conclude that group selection can't be dismissed a priori. And in fact, it's on a par with individual selection with respect to model selection considerations. In both cases, the question is, do you want to introduce a variable into your model, a parameter into your model that represents within groups variation, between group variation in fitness? The empirical argument in Williams' book is about sex ratio. He says that individual selection will produce an even sex ratio, and group selection will produce a female bias sex ratio when resources are abundant, and a male bias sex ratio when resources are scarce. And Williams then says that what we observe in nature is only even sex ratios. So he regarded this as a test case for group selection, the conclusion being that it ain't happening. But unfortunately, Williams was not aware that sex ratios in the social insects are often female biased. So that didn't work out so well as an empirical argument for the conclusion that Williams wanted to draw. In the context of a whole book that's devoted to attacking the idea of group selection, it's interesting to note a small part of that book in which Williams says that there is only one convincing published example of evolution by group selection, the tea allele in the house mouse. And he refers to this paper by Latent and Dunn, who actually followed Williams's parsimony protocol. They described the within-organism effects of tea, namely that in TW heterozygotes, W being the wild type, T forms 85% of the gametes. And individual selection, they took into account, taking into account that homozygote TT males are sterile. And they used those two facts about the T allele to predict what the frequency of the T allele should be in nature. And when they observed the house mouse, they discovered that the prediction was too high, which justified them in introducing the idea of group selection to explain why the T allele has the frequency it has. It's interesting that in this example, individual and group selection are pushing in the same direction. They're both selection against the T allele. And the puzzle for me is, if Williams thinks that this is a convincing example, why was he so willing in the rest of the book to be so negative about group selection? Maybe better constructed studies would show lots of group selection out there in nature. So it's a puzzle about this example, which I'm glad is in the book, but how it ramified. I don't think Williams sort of allowed it to ramify for the conclusions he was drawing globally. few years after Williams's book, Lewontin published this review article, and he used a single conceptualization of what evolution by natural selection means to characterize three possible units of selection, which David described, and I'll now repeat. Intragenomic conflict means that alleles in the same organism differ in fitness. Individual selection means that organisms in the same group differ in fitness, and group selection means groups of organisms in the same metapopulation differ in fitness. And this is multi-level selection theory in a nutshell, and it has a kind of pluralism to it in two different ways. First of all, a given trait can evolve under the influence of multiple levels of selection. So it's not like a single trait there's only one thing that's called the unit of selection in which it must evolve. And the second thing is that different traits can evolve with different units of selection. So it's not part of this picture that the only thing that matters is individual selection. The only thing that matters is group selection. Thanks.
Thank you very much for that, Elliott. Thank you. And just a quick note, because I know we had a lot more people come in after Elliott began speaking before we said all the info about this. If you wouldn't mind, if you have a question, go to the right where the three dots and raise your hand, and then we will call on people that way. And yeah, so we have about 7 minutes left for questions right now. So if people have questions, please.
And let's stop sharing the screen. Let's stop sharing the screen so we can see us.
Oh, yep. Sorry.
By the way, we have
Elliott,
82. We have 82 participants. Everyone, what a wonderful audience.
Yeah. I'll stop sharing.
Okay.
Please don't be shy. If you have a question or comment, then please raise your hand. And I think if you raise your hand in your screen too, then it automatically raises your hand. I don't know. I've heard that.
I would say good. We have Robert here.
Yeah, thanks so much. Fisher refers to the evolution of sexual reproduction as a possible exception to what is otherwise sort of what seems to us these days, for a variety of reasons maybe we can get into, but what seems to us to be a very genic orientation. Does that ring a bell? And would you say that maybe Fisher's comments on the evolution of sexual reproduction itself as a group level phenomenon might put him along on side of, say, you guys and
Well,
Darwin? Yeah, go ahead.
Yeah, you could ask the same question about Maynard Smith. I mean, their big picture is that group selection is 99% of the time a blunder, but there are these special cases that they feel like they can't be so dismissive about. So, and as like in Williams's book, he admits there's one case of group selection that you could actually believe in, the T allele, but that's a hiccup compared to the big picture that they really present.
And Robert, I'm going to attempt to answer you that exact question in a few presentations here. So definitely more on that to come.
Williams also, if I can add, eventually included disease, the evolution of virulence in disease, as he accepted as an example of group selection. But somehow this didn't penetrate to any kind of change in the general likelihood. And also Maynard Smith's model, 1964, so-called haystack model, was another nail in the coffin of group selection, or seemed to be. And one point to make is that before the advent of computer simulation models, and we have to remember that back in the 50s and 60s, there was not widespread computing, so people weren't knocking out computer simulation models. And so their resources for doing so were extremely limited, algebraic models. And so one reason why Maynard Smith's haystack model seemed to be so pessimistic about group selection were the simplifying assumptions he made, namely within group selection ran its course in each and every group containing both types. He had to make that assumption to simplify the math. And as soon as you can do a simulation model on the very same haystack model, which I did in 1987. So that was like over 20 years later. You could see that, au contraire, between group selection in the haystack model was a very tangible force, but 20 years later it made a big difference, and so it didn't change anyone's mind. So
Anybody else? Please don't feel shy. By the way, there is a Q&A function if you do want to ask a question, but you don't want to speak in front of everybody. Because by the way, this is being recorded, so just so everybody knows. But if you want to ask a question more in private, then feel free to put it in the Q&A, which is at the 3 dots below.
there we go.
Yes, Anna, please.
Hello, I'm Anna Lindholm, and I work on the T haplotype. And I wanted to ask you a little more about the evidence for group selection, because Lewontin and Dunn didn't know about all the fitness effects of the T. So they didn't think about for example, the sperm competition disadvantage that males have. And if you model this as well, then you can account for the T frequencies we see in nature.
That's a correction to what they were arguing. So maybe the bottom line now, would you say that the bottom line now is that there's no group selection?
I don't see evidence for group selection.
What I think they were thinking is that a group that contains all TT males is going to go extinct, and the females in that nest will have copies of T which will get dropped out as well.
It is theoretically possible for that to happen. But most T variants don't have sterile effects, but lethal. And then you can't lose the T. You can't get to that extinction point because there always will remain some wild types. So it might be a very specific part of that.
Thank you for that, Anna. I think we're gonna have to cut you off there. I'm sorry, because we have to go on to the next presentation. But I also implore you to stay around, because Elliott did a wonderful job of setting up the necessary historical context. But in particular, the next few presentations here will be on the empirical evidence for multi-level selection and group selection here. So without further ado, Joan, if you wouldn't mind sharing your screen and just introducing yourself.
Yeah. How does that look?
Good, good.
Okay, thank you so much, David and Mitch. My name is Joan Roughgarden. I'm a professor emerita from Stanford University, and I'm now an adjunct researcher at the University of Hawaii at the Marine Station. And I'd like to speak to you about some multi-level processes. I'm coming at this as an ecologist, and I'm interested in multi-level processes generally, one of which is what I would term canonical multi-level selection, as Elliott just described in some detail. And so this is where selection at the lower level leads to selfishness, but selection at the higher level favors cooperation. And if the selection at the higher level is stronger in a sense than selection at the lower level, then cooperation evolves in the metapopulation as a whole. And that's canonical group select multi-level selection, and it definitely occurs in some domains. It's the dominant process, and I think it's extremely important, and you're going to hear many more examples of this. And the talk that I give really could have been scheduled at the end of this session, but for scheduling reasons, it's up here in the front. And I want to talk about multi-level selection or multi-level processes involving holobionts. And just for terminology here, a microbiome, I use Josh Lederberg's definition. A microbiome is an ecological community of commensal, symbiotic, and pathogenic microorganisms that shares the, quote, body space. I love the phrase, body space of a host. And a holobiont then is a composite organism consisting of a host together with its microbiome. And Lynn Margulis authored this, was a strong proponent of this idea and did a lot to encourage its adoption. Now, a hologenome is the union of all the host genes with all the genes in its microbiome. And Elena Zilber Rosenberg and Jean Rosenberg really came forth with this idea and have been strong proponents of it. And a hologenotype is a configuration of the hologenome in a particular holobiont. And holobiont selection is differential reproduction and or survival of holobionts depending on their hologenotypes. And I have pictures of corals up here showing holobionts where the microbiome consists of the algae that are in the tissues of the coral and provide photosynthate to the coral. And actually, of course, on a coral, there are other microbiomes in addition to the algae. Now, the interesting idea about the hologenome is that the nuclear genome, the idea is that the nuclear genome can be augmented with the genes of the microbes, so that there's a composite genome that natural selection is acting on. And this is a diagram of how that might work. So these over here, so a hologenotype then consists of the genes in both the algae and the genes in the nucleus. And these are indicated by these dots here. And these, in the case of two host alleles and two microbial strains with horizontal transmission, you wind up getting 12 possible hologenotypes. In this case, it would be red in the nuclear part, but no algae. In this case, one algae that's green, one that's brown, and one that's brown and green. And so the way this works is that when the generation starts, the host releases gametes, the coral releases gametes. The gametes fuse to give you an array of juveniles over here through random union of gametes in classical natural selection on the nuclear genome. Then these larvae are colonized by Poisson sampling from a larval or from a microbial source pool. And Poisson sampling, just so that we're on the same page here, that's the sort of thing that you listen to with a Geiger counter. So if the radiation is low, you'd hear, if the radiation is high, you hear as particles strike the sensor. So in this case, we have the larvae over here being colonized at random by the larvae, by the algae. So you get and if the algae are very dense, then you get and so forth. So that's the underlying sampling process right here. Now, what's very interesting is the parallel between random union of gametes for this part, for the nuclear part of the hologenome, and Poisson sampling for the microbial part of the hologenome. So these are just parallel processes going on at the same time, which wind up generating these hologenotypes. And then then once the larvae have been colonized and are ready to grow, then the microbial community comes to equilibrium within the hosts, and then the hosts with their microbiomes are subject to holobiont selection. So the whole entity right here survives and reproduces depending on its genotype, its hologenotype. So that's the basic setup for a holobiont with a hologenome. And then the question is, can a process like this explain the main question, which is the degree of integration between microbiomes and the host? That's the thing, that's the topic, that's the main issue to be accounted for. How does it come about that the microbiome and the host are as integrated as we now know they are? And so I'm going to show you a simulation. I've done a lot of mathematics on this, but I'll show you a simulation which shows a difference in the way inheritance works for the microbial part versus the nuclear part. Now, as you know, in ordinary natural selection, if the genes involved are selectively neutral, then you get the Hardy-Weinberg law. And the initial variation continues unchanged unless a force comes in, such as drift or mutation or natural selection. But we'll see that there's no concept of neutrality for genes carried by the microorganisms. So this is an array right here. So what's happening? Okay, these dots are happening. So we have an array of 100 by 100 juveniles who are about to be colonized. And if they're colonized by a red algae or a green algae, or if they're colonized by both, they get a yellow right here. So these represent what's happening as through time as the holobionts are being colonized. And here we started out with an initial condition where most of the holobionts had red algae in them. But slowly through time, they fill up and come to an equilibrium gene frequency ratio. Now here, starting from the other side, where they're mostly green to begin with. And otherwise, these two colors are identical in terms of their impact on the holobiont survival and reproduction. So these are selectively neutral microbes. And we see that it looks about the same. And I have a little summary slide next, hopefully. Yeah. So here in this case, we started out with an initial frequency of the red at 0.8 and an initial frequency of actually 0.7, initial frequency of the green at 0.3, and they equilibrate at 50-50. And similarly, in the opposite direction here, they equilibrate. So that means that the underlying the underlying colonization process is structuring the outcome. And the upshot of this is here, that these represent the fitnesses of the holobiont, and if there are no microbes in it, its base fitness is 1. If there are microbes in it from type 1, which is red, the fitness is also 1. If they're, so to speak, heterozygous for both types, then the fitness is 2. And if they're homozygous for the green, the fitness is 3. Now, if you look at that fitness ordering right there, if something like this were taking place with nuclear genes, you know that you would fix right away on the most favorable gene. But in the case here, you don't necessarily fix on the favored microbe. So you only do fix on it if the colonization parameter is low. So this is the parameter that controls the density of in the Poisson sampling process. So if the Poisson sampling process is weak, and you just hear just a few hits now and then, you do wind up fixing on the favored gene. But if the colonization is high, then in this case, you actually wind up with a polymorphism. So that's D of 0.01 in this numerical example. This is D of 0.02, which is bigger. So that's the bottom line for selection on the hologenotype for the microbial component of the hologenotype, is that it's not as powerful a process as selection on nuclear genes. And in my opinion, it's not a strong enough process to account for the universality and strength of microbiome host integration. And so I hunted around for an alternative process that could account for microbiome host integration. So I'm going to show you another simulation right here. This is very amateurish. This is so this will involve a notion of adaptive assembly versus what I'm calling host orchestrated species sorting. So it's adaptive assembly, and the host controls who can come into it. And there are three types of algae right here, one of which contributes a lot to the host, so it gives a lot of photosynthate, one not so much, and one doesn't do anything. And furthermore, the algae can be classified by what their within host carrying capacity is. So they could be low or they could be high. And here's where the host selection comes in. The host can, if the microbe doesn't contribute anything, then the host views it as a pathogen and activates its immune response, and that's what these little tanks are supposed to be, and they kick it out. And if it's only marginally helpful, then the immune response is weak, and if it's really doing a lot for the host, then the host welcomes it and doesn't kick it out at all. So what happens? To begin with, this consider one with a low carrying capacity. So it goes in, it tries to go in, but it doesn't have enough carrying capacity to give the host anything. And so it doesn't enter, and the host doesn't do anything for it. On the other hand, if the algae has a high carrying capacity within the host, then it enters but is rebuffed. And if the host has and if the algae contributes a whole lot to the host is glad to get it. And then, however, if an algae comes in which doesn't give as much to the host, it then is able to exclude the most altruistic algae, and you're left with a configuration in which you do have integration. So you have a microbe that is contributing to the host, but not as much as it could, and you're getting an immune response to it, but not as bad as it could be. And so you get this net configuration here. So that is an integrated, not an optimally integrated, you might say, but that is an integrated microbiome host situation. And I'm going to suggest that this is the main process that would account for host microbiome integration. Now, this is not really, this idea of adaptive assembly, I think, may be independently being conceived in a number of different domains. Just last week, I discovered these two papers right here, plant-driven assembly of disease-suppressive soil microbiomes, microbiome assembly in fermented foods. So we're looking at this issue of an alternative to a selection process being an assembly process. And so on the basis of that, I think we could change or amend the definitions of microbiome accordingly. Instead of using the phrase body space of a host, as Lederberg suggests, we could use something like this, which is a microbiome is an ecological community that occupies the neighborhood controlled by a host. And then a holobiont would then be, instead of focusing on them as an organism, they're a functional unit, and they are manifestly a functional unit, consisting of a host integrated with its microbiome. And I think that this would then move us forward for how to understand how these things form. And then the references on this are, so I have 3 key papers on it, which are right here, in case you want to copy them down. So that's what I have to say, and thank you very much.
That's awesome. Thank you.
Thank you, Joan.
Really awesome. Let's get back onto the gallery, and we have actually a good long time for questions here.
So I need to de-share, right? How do we do this?
Just at the bottom where it says share screen, if you click on the, I think it should say,
Yeah,
maybe up top. Yeah,
Uh,
top, it says view options and
just
stop
stop
participants.
share.
There you go.
we-
There we go.
Perfect. Yeah, so
Okay.
please ask a question.
Raise your virtual hand.
Mhm.
Errol. And please briefly introduce yourself while you're at it.
Hi. Hi, John.
Hi,
Great
Errol,
talk.
how are you?
Good to see you. So I wanted to ask about, just wanted you to tell more about sort of how that sort of carrying capacity and evasion interact with each other. Is it, do you assume like a Lotka-Volterra type
Yeah,
competition model inside the host?
yeah, the within host dynamics are Lotka-Volterra.
And so then the competition coefficients between the symbionts, like how are they determined?
Well, it's assumed that the one that gives less to the host then has a higher carrying capacity in this case. But I think the competition coefficients were arbitrarily set at 0.5 or something like that. So it was being driven by the carrying capacity difference, not the competition coefficient difference.
Okay, I see.
Yeah.
Okay. Yeah, that was my, I just wanted to hear more about that.
Yeah, because it's the algae is giving up of its carrying capacity to help the host, because it's giving photosynthate that it could have used to grow a higher within host population size, and instead give that to the host. But it benefits. It's a multi process, because it then benefits when the host benefits as well. So it does receive a benefit from that. But it's because it's primarily an assembly process that accounts for the integration that occurs. And but there's still selection going on, but there's unilevel selection going on in endowing the host with the capability to do this sorting, to do this admission process. So unilevel selection is accounting for the evolution of the immune system, for example.
Right.
And you needn't have, my idea of the sorting was going through the immune system, but you could also have incentive-based systems instead of punishment-based systems, which is what the immune one is. What do you know?
Thanks.
Yeah.
Elliott.
Excuse me. I understand the idea of how the host is the selective agent, as you said, governing the admissions process. But why isn't there also, after the admission has taken place, selection on the entire holobiont as a unit?
Oh,
I
there
don't see
is.
why. Oh, so I misunderstood you, because I thought you were saying that's out, and all we need to think about is the selection process that the host is controlling.
Oh,
That's not your view.
Yeah, I'm sorry, I was unclear about that. Yes, the selection process on the host as a whole, no doubt about it. But the problem is that and that one slide I had in there showing you that selection only was effective if the colonization coefficient was small compared to large. There's selection on the host there. The problem is the selection on the host is fighting against, if you will, the influence on the gene, on the microbe frequencies from the colonization process. So you really get an appreciation of the value of Mendelian inheritance when you study this, because the Mendelian inheritance preserves the notion of neutrality for the alleles if they're neutral with respect to fecundity and survivorship and so on. But here, because there's no notion of neutrality at the microbe level, even if you do have selection at the holobiont level, it's fighting against the colonization process. And in addition to whatever within host selfishness might be going on. So this colonization process then winds up throwing a monkey wrench into the analogy of microbes as just being an extension of the nuclear genome, which is a very appealing idea. And I spent quite some time working this through. A lot of the objections to the holobiont, to the hologenome concept in the first place, I don't think were well founded. And I think they were kind of a priori and negative and so forth. But this one about the absence of selective neutrality, is really pretty fundamental and hadn't been raised by any of the people just making verbal complaints about the whole idea.
Mm-hmm. Thanks.
Yeah, so I have a comment followed by a question. The comment is that there's no question that for many kinds of interactions, the size of the holobiont community and the degree of horizontal mixing would not be favorable for that as a level of selection. You wouldn't have variation because of there's just too much mixing. But when you actually do experiments, you find much more variation than expected on that basis. And we've talked about this, so that's why I'm making it a comment, not a question. And I think what's going on, and Michael, in his talk coming up, I think is going to be addressing this with laboratory experiments, is something closer to Wright's interdemic selection. In other words, multiple equilibria, which, and you're selecting among multiple local equilibria, which I think provides a different basis for evaluation. But my question is this: within your own models, it seems to me, given all the horizontal mixing that takes place, then when we think of the host acting as some kind of engineer, which it does take place, I mean, that's a powerful idea. And so I think that it will be the case that the host organism is doing a lot of engineering of its own microbiome, but it can't just surgically go in and work with individual types. It's going to have the same problem that a human experimenter does in terms of when it has an immune response. It's going to be eliminating whole communities, not single types within communities. So I feel that the host as engineer concept is going to be facing some of the same multi-level problems that you've identified for holobiont evolution. Take, for example, a legume plant with nodules, and the nodules are being colonized by different bacteria. Well, it can abort a nodule, but it's by doing so, it's aborting an entire community, not a single strain of bacteria. So there's my question.
Well, I mean, that's the limitation of the modeling so far, is that it's focused on a single strain or two genotypes within a single strain. And insofar as the impact of the microbiome on the host is via a community, not just a collection of individual bacteria or algae, but if they're working as if the microbiome is working as a consortium, then yeah, I haven't looked into that, and therefore then the immune response would have to be to a consortium or something of that sort. So, so off the top of my head, I don't know quite what to say about that. That's a good point.
Okay.
So, Joan, there is one question in the Q&A. I don't know if you have time, if you want me to read it, or if you want to just answer it in private.
No,
If
go ahead and read it. Sure.
Okay, yep. So it's from Eva. She says, "Thank you, Joan. In your microbiome colonization example, are you taking within host growth and competition into account? I'd expect that to tip your results away from neutrality."
Yes, I'm definitely taking within host competition and growth into account. And as I was mentioning to Errol, the within host community dynamics are Lotka-Volterra competition equations. And so that's built into the model. So that's already there. And so these results apply even though that's present.
Okay.
Awesome. Well, thank you again, Joan. And so we will move here to Michael Wade. Mike, if you wouldn't mind introducing yourself and sharing your screen here.
Okay. My name is Michael Wade. I've been studying multi-level selection for about 50 years, not counting my work as a graduate student. I would extend it to maybe 54 years. And today I'd like to talk to you about interdemic selection in metapopulations, and with an emphasis on the question of populational heritability. And before I get started, I want to thank David Sloan Wilson and Mitch for organizing this symposium. I think it's just a great opportunity to bring all of these different ideas together. And I think the success of a theory of multi-level selection can be judged by how it can be applied to sort of the more recent collectivities like holobiomes, as in the previous talk by Joan Roughgarden. I think there are some really brilliant ideas there. And I think if we can integrate these as a community, I think it would be terrific. In any event, let me talk about some experimental work on interdemic selection. So interdemic selection takes place within a metapopulation, which I'm going to use the definition of a metapopulation from Richard Levin's collection of local demes of differing size and genetic composition, each subject to potentially different forces of local selection and connected by varying degrees of migration. You might depict it, as I've depicted it elsewhere, something like this, where each circle represents a deme. The size of the circle represents something about the age of the deme, whether it's close to having just been colonized or whether it's a long way from the colonization process. Sometimes demes go extinct, sometimes they get colonized, and there's migration back and forth among these different demes. Now, interdemic selection was proposed as an important feature of Sewall-Wright's shifting balance theory. Sewall-Wright derived his theory from his studies of the domestication of cattle breeds, which he found that that process of domestication involved two levels of artificial selection. The first level of selection occurred within small local farms, where each farmer worked to improve his herd, culling those that deviated from some sort of idealized norm of the breed and breeding from those closest to the norm. But local selection on small farms was limited because artificial selection, starting with small numbers of breeding cattle together, lead to fixing very quickly of local genetic variants by a combination of drifted selection. The second level of selection that Wright could detect and document from the pedigree records of domesticated cattle was selection among herds for sires. Furthermore, he found that among herd selection for sires was responsible for larger increments of trait change than within herd selection in shaping the overall characteristics of the breed. In his own words, Wright said that his studies clearly showed the importance of selection at two levels. One was selection among individuals and building up herds, selection favoring an allele at each pertinent or relevant locus that gives a favorable effect on the average of all the combinations, and he called that genic selection. Then the breeder selection among herds as sources of sires was selection among the diverse interaction systems that each farm had happened to have arrived at. And he called this organismic or genotypic selection. And he recognized that the two-level process was much more efficient than mere individual selection, and it led him to consider whether an analogous two-level process might occur in nature. So much like Darwin used artificial selection and successive breeders as a model for his theory of evolution by natural selection, so too did Sewell Wright use his more detailed genetical analysis of artificial selection by cattle breeders as a model for his shifting balance theory. Wright's famous conceptual illustration of a metapopulation looks something like this, where these are contours of mean fitness, and the little pluses indicate fitness peaks, and the little minuses indicate fitness valleys. In our hands, myself and Charles Goodnight, we envisioned Wright's metapopulation as something like this, in a wonderful graph produced by one of my doctoral students, Jeff DeMuth. And in our hands, we create little tribolium flower beetle populations. Those populations, one way or another, end up at different fitness peaks, like the little pluses on Wright's diagram, and there are little fitness valleys separating these peaks. Like Lewontin, we envision, and like Wright, we envision that metapopulation properties are necessary for adaptation by interdemic selection. First of all, there has to be phenotypic variation among demes. And that whatever that phenotypic variation is, that has to cause fitness variation among the demes, which is the source of interdemic selection. But you can have all the selection you want, and if there's no populational heritability or heritability of the population mean, then you're not going to get any response to interdemic selection. And the heritability at the group level has been one of the more controversial properties. Elliott hinted in this by pointing to Williams' first a priori argument, saying that because evolution is change in gene frequency, the gene is the most fundamental unit. How in the world could a group of genes in an individual, let alone a collection of genes across a population of individuals, or in the case of that Joan Roughgarden just presented, let alone a collection of multiple independent genomes across multiple species, have any property of heritability? So the phenotypic variation with flower beetles just comes naturally. You set up a bunch of populations with a fixed number of breeding adults, and they grow to different sizes spontaneously because of all kinds of things they do to one another in the flower. the mechanisms of interdemic selection that you might impose on an array of metapopulations of flower beetles, there's a variety of things you could do to cause interdemic selection. You could look at differential dispersal of migrants out from some demes and into others, as in right-shifting balance theory. You could create a differential fissioning of demes, as happens in some groups. You could do differential extinction of local demes, where some demes persist longer than others. Or you could have differential colonization by local demes, where colonizing propagules originate more often from some demes than it does for others. There are lots of different types of fitness variation among demes that could lead to interdemic selection. In the diagram that I showed, on the left, we have an experimental population where the large demes are sending out migrants into established smaller demes and also sending out larger areas to colonize smaller demes. In our experiments, one deme tends to be a group of 20 adult beetles in 8 grams of flour mixed with yeast medium. And those founders, over a period of 30 or 40 days, mate, lay eggs, hatch, eat each other, and develop and become a much larger number of offspring. The metapopulations that I'm going to talk about here consisted of 50 demes. And if they're founded with 20 adults per deme, the starting situation at the beginning of every generation is 1,000 adult beetles. Pretty decent total population, although it's subdivided into 50 separate demes. One of my favorite features about the experimental design that Charles Goodnight and I came up with to study interdemic selection was the feature that in the first generation of our studies, the middle generation, the orange one, the 50 demes were established with no interdemic selection, no random migration. They were just established from the initial stock. It wasn't until we were able to count the numbers of offspring in those demes that we could then treat them differently and create an experimental metapopulation. And by doing something just a little bit different, create a control for that experimental. What I find attractive about this design is that deme 1 in the experimental was the same as deme 2 in the control. Deme 2 in the experimental was the same deme as deme 2 in the control, and so on at the start of the experiment. It's really kind of a nice paired design there. How did we impose interdemic selection? Well, we simply census the number of offspring produced by the 20 initial adults in each of the different demes. And we could assign a deme then a relative demic fitness by taking the numbers of offspring that the group of 20 had produced and divide it by the mean number of offspring that all 50 groups of 20 had produced. Assigning relative fitness to the deme in this way means that demes with relative fitness greater than 1 are going to send out migrants, and demes with relative fitness less than 1 are going to become recipients of migrants. How did we set up the next generation? Well, you simply multiply the number of founders for each deme 20 by the relative fitness of the deme. This guarantees that the total number of founders is going to be 1,000, but some demes, those whose relative fitness is greater than 1, will send migrants, and those whose fitness is less than 1 will receive migrants. The senders pool their migrants and we distribute them into the receiver demes as needed. What's the control for this type of differential dispersion of migrants out of demes of high fitness or high productivity into demes of low productivity? How do we create a control metapopulation? Well, we decided that we wanted the control metapopulation to have exactly the same numbers of migrants moving between demes as our experimental populations. So we counted up how many migrants dispersed out of the demes with a fitness greater than 1. Then we take the number of demes, 50, choose them at random and with replacement m times, and the numbers of times deme I was on the list is how many migrants it gets or how many migrants it sends. So a deme could send a lot of migrants but not receive any. It could send as many as it received. It could do almost anything. But the important feature is that the total number of migrants in the experimental metapopulation equaled the total number of migrants in the control metapopulation. Therefore, on average, the number of migrants per deme was identical. The difference between the two metapopulations was that in one case, migration was differential, associated with producing a lot of offspring, and in the controls, it was random. We had 3 of these experimental control metapopulation pairs. We had a total of 6 metapopulations in our experiment. And I'm only going to talk about the results of 2 of them. In one, we did this differential migration process for the experimental population at every generation. And of course, we had a control where we had random migration in exactly the same amount at every generation. And in a second experimental control metapopulation pair, we did the differential migration only once every 2 generations and random migration only once every 2 generations. In the generation in between, we just took 20 adults out of everybody, regardless of how productive they were. Let's get back to the notion of populational heritability. So I just explained how we imposed interdemic selection. How do you measure populational heritability? Well, you can measure the realized populational heritability, which is the regression of the response to interdemic selection on the cumulative interdemic selection differential. This concept of realized populational heritability is identical to the one used by breeders who select and choose their characteristics by measuring individuals. It's a modification of that to apply the concept of realized heritability to a metapopulation instead of to a population of individuals. The response to selection in our case is the mean of the experimental metapopulation minus the mean of the control population divided by the standard deviation of the variation of populations from one controlled deme to the next. The interdemic selection differential is the mean of the parent demes minus the mean of the experimental demes before we impose the interdemic selection. The same way you define a selection differential at the level of the individual. And again, that was standardized by the standard deviation in the experimental population. This is a graph of the cumulative interdemic selection differential on the x-axis versus the average standardized response to interdemic selection on the y-axis. The y-axis goes up to 2.3, meaning that there were 2.3 standard deviations more beetles per deme in the experimental metapopulation than there were in its control. There are 2 lines on this graph, one through a bunch of solid circles and one through a bunch of empty squares. The black dots represent the realized heritability, the selection differential divided by, or the response to selection divided by the selection differential for the metapopulations where we impose these processes at every generation. And the empty squares represent where we impose these processes every other generation. And you'll see that the realized heritability, when we selected at the group level, every other generation was 5-fold larger than the realized heritability when we selected at every generation. This fivefold difference in realized populational heritability suggests that there's some kind of non-additive or epistatic genetic basis for the response to interdemic selection on mean fitness. And couldn't there be a better way, rather than just look at the end of the process of the realized heritability, couldn't there be some better way of trying to see whether there's a non-additive genetic basis to mean fitness? Well, we have some guidance from additive theory. This is from a table of Falconer's book on Introduction to Quantitative Genetics, is table 15.1. It tells you the relationship between genetic variability and some trait measured on individuals and how that is affected by the population structure. So within a deme or within a population, the additive genetic variation from one individual to the next in whatever character you're measuring goes down when there's population structure. It's diminished by a factor of 1 minus FST. And where does that variation go? Well, it goes between groups, and it shows up in differences from one group to another in their group mean. And that difference should equal 2 FST times the original additive genetic variance for this trait that you're studying. And FST is Wright's measure of the heritable variation, one gene for one gene that is among groups. Tragedy for those who study interdemic selection, and the question that David Wilson brought up in relation to Joan's experiments, or Joan's theory, is that FST plummets with any kind of migration between demes at all, where n times size of the deme times the migration rate, nm, which is the x-axis here, is the number of migrants that move between demes. So how from a tiny value of FST can you ever get any, first of all, the variance on which to select, and secondly, any kind of heritable variation? So as a consequence of the additive assumption, what I'm saying is that the genetic variation among individuals in whatever trait you're measuring is much, much greater than the variation among demes in the mean value of those characteristics. Well, let's actually do this with flower beetles. Let's take an ancestral stock that happens to be segregating for a black body color mutant and a wild type mutant. It's got some variability in fitness of those beetles. And let's subdivide that population for 13 generations at a deme size of 25 founders. Each deme reduced to 25 founders every generation for 13 generations. Drift creates variation in mean fitness, or a variance among demes in mean fitness during this process. Where does this variation come from? What does it say about the origins of the variation, that little v of w sub I sitting inside the ancestral population? Well, we know how to measure that too. We take the ancestral population, we take out a bunch of males, we take out a bunch of females, and we construct a half-sib design, where by crossing males to randomly selected females, we can estimate the additive genetic variance for fitness within a deme. Well, we want to do this in a populational context. So what do? Is we combine this design with this design and get this. At the top, we replicate genetic individuals in the way suggested by quantitative genetics. And down the row, we replicate gene networks in the way by making multiple replicates of these different demes. Each deme now is going to be founded in a way that's different from the experiment that I just showed you. It's going to have one male or female from the top design and 15 adults from the deme over on the left. when you put a marked male, a male that's homozygous for a black body color, in with a bunch of wild type adults, that allows you to count or estimate the individual's fitness by simply counting how many adult offspring that it produces within a deme. It lets you see within a deme at least what one individual is doing. And then, of course, the quantitative genetic design lets you convert the variation in these numbers into the fitness variance, the additive genetic fitness variance for the individual. So the question we're asking, what's the magnitude of the variance in fitness between individuals in the ancestral population and the variance in fitness among demes on which we measured the realized populational heritability? We know, theory tells us that the variance among demes should be 2 FST times the variance within the ancestral population if fitness is additive. We also know that FST is about 0.15 because we measured it in jillions of other experiments done with these same stocks of beetles. So the additive expectation is that the variance in fitness among demes should be 0.3 times the heritable variance, additive genetic variance in fitness within demes. What did we observe when we actually ran the numbers on the experiment that I just described? Well, we found that the variance among demes was 18 times the variance, the additive genetic variance between individuals within demes. That's kind of a big miss for the additive theory, because that's 54 times too much variation between demes to be accounted for by the variation in fitness between individuals in the initial stock. So therefore, one of the inferences you could draw is that fitness appears to be non-additive. Now, this probably wouldn't have surprised Wright, because he stated many times throughout his career that describing selection in terms of single genes was very inadequate, and that quantifying population genetic structure with his measure of it, FST, was also wholly inadequate, because FST describes only single genes and not gene combinations. So in summary, we found that mean fitness in all the experimental populations increased in response to interdemic selection by differential migration. We found that the realized heritability with interdemic selection every other generation was 5-fold more effective than imposing it at every generation. We found that a fairly large response to interdemic selection, despite moderate FST of 0.07 by Wright standards. Now, don't get confused by my having shown you an FST of 0.15. That was for the meta population where N was 25 for 13 generations with no migration. This FST of 0.07 is from the control meta population during the shifting balance experiment. So fitness appears to have a non-additive genetic variance, which allows interdemic selection to be much more effective than it is expected to be under additive theory. And I'd like to thank my career-long collaborators, Dave and Charles, who both passed, and many wonderful continuing conceptual and empirical and theoretical collaborators, Jim Griesemer, Jake Morad, Jason Wolf, Robert Skipper, Felix Brayton, and Lisa Lloyd. And I'd like to thank the NIH for giving me enough funding to get to the end of these experiments, although sadly they wouldn't give me any more to continue studying these phenomena. And that's 25 minutes exactly.
Thank you so much, Michael.
Wow. So that is amazing. You know, I can't resist being the first to ask a question, Michael, because it seems to me that the distinction between an individual level trait, a phenotypic trait that you measure in an individual beetle, and a trait that might count as social, that distinction dissolves completely, because the individual level trait is so much a matter of genetic interactions. Would you agree with that, or is there any useful distinction between a trait that you can measure in an individual and a social trait?
Well, we know because of the early experiments that we did where we used Park's methods to measure individual characteristics like how many eggs a female laid, how long developmental time was, how much cannibalism there was, and things of that sort. And we were not able to explain the results of our early group selection experiments, which were kind of brute force differential extinction and colonization without any migration. Later on, Dave Macaulay was the one who suggested that maybe we set up one of these sacrifice experiments, and we could see then that characteristics of individuals in groups were not at all the same as characteristics of individuals separate from their groups. And in this experiment that I showed today, we actually had some way to measure what went on between individuals within groups, at least in terms of how many offspring they produced that contributed to the, the mean of the offspring that we're counting when we census the populations in our metapopulation experiments. And you can see that the heritable variance in that trait, which is probably most relevant to the group mean, really doesn't explain at all the variance between means, at least not under additive theory, it doesn't. So yes, I can agree completely with you. And, I wish I could say more. I wish I could pin it down with using molecular genetics or something like that to gene combinations the way Wright would have liked, although I'm not at all sure that it would, a simple, nice, understandable pair of genes would pop out like in the simple two-locus models.
Right.
So.
Any other questions here? If you wouldn't mind just raising your hand. We have about 1 minute, so time left for just one quick question. Okay, sounds good. Oh, Elliott, yep, go ahead, please.
Yeah, Mike, this question is on my mind. I don't know that it's directly relevant to the studies you were describing, which were excellent. There is this theme that I've been discussing with Ford Doolittle about this objection to holobiont level selection, in which he says if there are too many parents of an offspring population, that you can't get a response to selection at that level. Do you have a reaction to that idea, that objection? It doesn't need to be about holobionts. I mean, just imagine an experiment in which a given daughter population is composed by taking n potential parent populations and sampling from those, so that each population has comes from n parent populations for some value of n. And the thought is, when n is too big, you don't get a response.
Yep.
Yeah,
I'm sorry, Michael, I'm going to have to cut off because we are at 6:30 here, but if you wouldn't mind, if you do have an answer to that, just put it in the chat for everyone to see there. So it gives you some time to think about it. But I would like to move next to Athena. And so Athena, if you wouldn't mind sharing your screen and also giving us just a little introduction on yourself.
Absolutely. Well, first of all, thank you so much for having me. It's a real pleasure and honor to be part of such an amazing assembly of phenomenal scientists. And I'm delighted to have the chance to talk to you guys a little bit about some of my work on evolution and cancer. So I've been working on the evolution of cooperation for a very long time. I got interested in understanding, how cooperation evolves and what keeps it stable as an undergraduate. And it's quite fitting, actually, that Michael Wade, you just spoke, because as an undergraduate, we were corresponding, and I, really enjoyed having the chance to talk with you then, and continue to later. And David, of course, we've been corresponding for a long time, and I remember you encouraging my early computational work on the evolution of cooperation, again, when I was just an undergrad. So it's wonderful to be able to be here with both of you and the rest of the community as well. So today I'm going to talk a little bit about how cancer and understanding the evolution of both cancer and our abilities as multicellular organisms to suppress cancer are a really excellent example of how the tensions between levels of selection can manifest in biological systems. And I'll present work that's sort of both about what happens at the level of cancer evolution within the body in terms of cellular evolution, and then and organismal evolution and how they sort of interact. So the story is fairly simple in terms of the big picture, that when it comes down to it, our multicellular bodies, within our bodies, selection at the cellular level, right, of the population of cells in our bodies will favor cancer, but selection at the organism level, so you have a population of organisms, will favor cancer suppression, right? The organisms that are better at suppressing cancer will do better in the evolutionary process. And so rather than thinking here about, you have an organism and then a set of organisms, we're thinking about, you have cells and then you have sort of sets of cells that are functioning at the organism level. And oftentimes we sort of think, well, an organism is sort of this unitary entity and we don't really need to think about what's going on within it in terms of the evolutionary process. But the work that, I and others have done over the last few decades on cancer as an evolutionary process has very clearly shown that what you have happening inside the body just over the course of normal aging is indeed an evolutionary process. And that's at the heart of why we get cancer. So if we want to understand really what cancer is, we have to go all the way back to thinking about the origins of multicellularity itself, and thinking about multicellularity as essentially, the evolution of multicellularity as the evolution of cooperation among cells. And essentially what happened is that aggregations of cells varied, in their level of cooperativeness with each other, their ability to engage in what we call the 5 foundations of multicellularity. So inhibiting their proliferation, controlling cell death, allocating resources as needed, dividing labor, and then creating and maintaining an extracellular environment. And those groups of cells that were good at doing those things were able to outperform those who weren't. Now, I see that there is a raised hand, but I can't actually go to view a question. Do you want me to take questions during this, or should I wait until the end here?
No, sorry, César, let's save our questions for the end, please.
Okay, great. So thinking about sort of what happened during the evolution of multicellularity, that was itself a multi-level selection process where within groups, of course, you could have cellular cheaters or cells that were sort of just acting in a way that might benefit them as an individual, but not necessarily benefit the group. And then you had other aggregations that had cells that had these features. And it was because selection favored those groups with greater cooperation that we even got the evolution of multicellularity in the first place. And if we look at what cancer is, we can see that it maps directly on to the breakdown of multicellular cooperation in these 5 foundations of multicellularity. So these are the 5 foundations that we've already talked about. And this is what we see in multicellular aggregations, from the smallest ones to the largest ones like us. But what we see with cancer is that each of those is compromised. So you have a disinhibition of proliferation. Cells will avoid cell death. You have monopolization of resources. Rather than division of labor, you have cells that shirk their duties and also co-opt the labor of other cells for their own purposes. And rather than creating and maintaining a shared environment, what you see is actually massive sort of environmental destruction in the form of the creation of acid that actually breaks down the extracellular environment that both sort of destroys the shared environment of the cells, and it also eventually makes it possible for invasion and metastasis, which are extremely important clinically when it comes to cancer, because those are the things that really threaten us as, multicellular organisms, as humans, and cause the most problems for people. So now that I've been talking sort of a lot about this idea of cheating, oftentimes people ask, well, what do you mean by cheating? Obviously, the cells are not consciously deciding to break the, rules of engagement. And so for the purposes here, I'm really just defining cheating as a breaking of shared rules that are leading to a fitness advantage for the rule violator. And that could be, in some cases, right, you have systems where it is conscious, but you can have those rules be completely implicit. And in this case, those rules are really shared rules that are encoded into the genome of multicellular organisms, where they're, they have rules for proliferation, inhibition, and sort of following all these rules of multicellularity. And it's mutations that break those, as opposed to any sort of purposeful process. So, and mutations, quite central and important here to understanding why you get the evolution of cancer, and also sort of the balance between selection within the body for cells that are cancerous versus selection on organisms to suppress cancer. So the higher the mutation rate, the more likely it'll be that any of these adaptations for multicellular cooperation get compromised because of those mutations. And so when we look at sort of, what can happen in a multicellular body, all of these foundations of multicellularity can get compromised, and the cells that are not following these sort of proper rules for multicellularity can then lead to the initiation of cancer or accelerate cancer progression if cancer has already started. So let's kind of pull all these pieces together before we go on to the next part. So basically, we have a situation where cooperation evolved in order to make it viable for multicellular aggregations to exist. And those multicellular aggregations, because of cooperation, were able to outperform, groups that were less cooperative. They may have avoided predation better. They may have been able to engage in behaviors that other, aggregations couldn't, or single cells couldn't. And so multicellularity itself is, cooperation. Then you have the ability for cells to sort of break down these foundations of multicellular cooperation. And if we bring it all together now, we have on the outside, so the inside, this is sort of the framework of the foundations of multicellularity and the breakdown of multicellularity that I developed with a bunch of colleagues around evolution and cancer. And then we matched this on to, on the outside, what we have is the hallmarks of cancer framework. This is sort of the accepted framework for what are the phenotypes associated with cancer in clinical settings when you actually look at, what characteristics cancer cells have. And what we found is that they really map on to these foundations of multicellularity and the breakdown of those foundations of multicellularity. Now, 3 of them have to do with uncontrolled proliferation, and there's sort of different mechanisms by which that uncontrolled proliferation can happen. 2 of them have to do with avoiding cell death. Again, 2 different sort of ways that can happen. Several of them have to do with monopolizing resources. One of them is sort of related to environmental degradation in terms of making metastasis more likely. But then we also found that there was not actually a hallmark around dysregulated differentiation, but this framework that we put together suggests that maybe that should be. And in fact, now over the last few years since this paper was published almost a decade ago, the community has been coming around to this idea that dysregulated differentiation is also an important hallmark of cancer. And so the fact that there's a sort of convergence between our kind of first principles approach using trying to understand the evolution of cooperation and then the breakdown of that cooperation with cancer evolution, and then mapping that on with the hallmarks, the fact that many of these things overlap, and that also this framework kind of suggests new ideas that haven't been considered in the cancer community was really reassuring for us that we're sort of on the right track with how we're trying to connect all of these dots. So let's talk now a little bit about how cancer as this sort of evolutionary process is manifesting across multicellular life. So we sort of talked about already how evolution among cells is inevitable. And basically what this means is that any multicellular organism is going to be susceptible to cancer. Including, this is one of my favorite examples. This is a crested cactus, and it is a cancer-like phenomenon that occurs in plants called fasciation. And as you can see, there are some really interesting features here. Not only is it sort of possible to see the way that the proliferation has been altered, you can sort of, see the growth formations in a way that you can't with a tumor in an animal, but it's also quite striking and beautiful. And many of these crested formations, whether they're in cacti or other forms of fasciation in other plants, are also quite striking and beautiful. And so I won't have time to really talk about this much today, but we've used crested cacti as a way to communicate with broad audiences about cancer and about cancer across life because of these features that make it easy to kind of see what's going on, and also they're quite lovely to look at. So I had the pleasure of spending a whole year at the Wissenschaftskolleg in Berlin with the Cancer Evolution Working Group, where we sort of had some time to sit down and talk about what we thought the big questions were in cancer evolution. And we spent actually a few months talking about just, what needed to be done, what the next sort of steps were in the field, and eventually decided that the first thing that we wanted to tackle was looking at cancer across the tree of life. And so the next few slides that I'm going to show you are part of a big collaboration that involved many researchers. I think we had about 10 or 12 people, all included, looking at what the existing literature was. Now, since that time, the field of comparative oncology has expanded a lot, and I'll show you also some results that we have that are more recent, but this will give you kind of a nice overview of just how cancer does exist across the tree of multicellular life. So if you look in animals, again, this was from our review of the literature, we found that cancer was observed across all of these different branches of animals, including in the earliest branches of metazoans, and that a loss of differentiation is something that is also observed in coral. In fungi, you see a loss of tissue differentiation, which is a sort of cancer-like phenomenon. You might not say that it's full-on cancer because you don't have invasion or metastasis, but you do have these cancer-like traits. And in general, the green algae. So this is what I showed you already, really, but just to expand the example, that the lower picture, that's actually a pine tree that has almost its entire soma is this crested trunk. So you can get these very interesting formations. And there's also been the observation of things that look like cancer, so bacteria-free metastatic growths. But this is still an area that's, where much more work is needed to understand, are the phenomena that are happening in plants directly analogous with cancer, or do they only offer some similarities in terms of the dysregulation of proliferation and cell death, but perhaps not all of the other features. So the bottom line here is that multicellular bodies have evolved to essentially be able to detect and then appropriately respond to the breakdown of cellular cooperation when you have cellular cheating, because selection acted at the organism level. And when we talk about cancer suppression, that's essentially what we're talking about, is selection that acted on populations of organisms to favor cancer suppression. So what are some of the ways that this occurs in terms of mechanisms? Well, within the body, you can sort of break it down into three main types. You have mechanisms that are happening inside the cell. You can almost think of this like I use the analogy of a cellular conscience, right? So it's the cell monitoring itself, checking its own gene expression, checking what it's producing itself. Is it actually contributing to the viability of the organism, or is it compromising it? And you have the neighborhood level. And so this includes monitoring cell adhesion, tissue architecture, basically, are things normal in the neighborhood? And then you have system-wide. So this includes the immune system, looking at, are there areas in the body where things are happening that are not normal? And if so, a response can be made to try to sort of deal with that in a more targeted way. And so all of that allows for the suppression of cellular cheating, because you have all of these sort of different mechanisms from all these different sides acting together to try to maintain that cellular cooperation. Let me give you one more specific example, which is TP53. It's been called the guardian of the genome, but I like to call it the cheater detector of the genome, because really what it is a central node in an extremely complex information processing network that is integrating signals that are coming from many different places and then sending signals out. And it's essentially deciding whether a cell is rehabilitatable or whether it needs to remove itself from the body in order to essentially save the body. So we can look at this as really a system that is taking a hugely complex set of information, both from inside the cell and outside the cell, and then using that to sort of make a decision about what the cell should do. Now, how is TP53 actually detecting this? Well, it is essentially producing more of the P53 protein if the combination of signals and cues that it gets suggests that what is happening within the cell is not actually good for the organism that it's in. And now, anybody who's seen these sort of network diagrams of gene expression, right? It's just impossibly complex looking, and that's because what we have essentially is a hugely complicated information processing circuit with every one of these cancer suppression genes that is taking in so many different kinds of information. And I'll also just add that the kind of decision that TP53 or any cancer suppression gene is having to make is basically, given an ambiguous set of information, is it okay for this cell to stick around? Or does this cell need to be removed from the body in order to protect it? And there are a lot of trade-offs that come from eliminating too many cells, right? So you can't just be like, oh, let's get rid of this cell and this cell and this cell, because then you actually have to replace those cells. And even just the act of dividing increases the chance of mutation. So there's no perfect way to do this. It's all a set of compromises that everybody has to make. And there's no way to sort of fully eliminate cancer risk because of many of these trade-offs. I'll share a few more things and then I'll leave time for questions because I see that we already have a few. We know from research that myself and colleagues have done that there are changes that have happened in the frequency of the TP53 gene over evolutionary time, and that when you have more copies of the TP53 gene, there's actually a more sort of robust response to radiation. So cells are sort of more likely to kill themselves, basically, to protect the organism. And you might be aware of this sort of work that elephants actually have more copies of the tumor suppression gene TP53. And that's most likely because being larger and having a longer lifespan requires more cancer suppression. Now, being larger means a larger population size, right, of entities, and a longer lifespan means a longer time that cellular group needs to exist. Right, so it makes sense that you would need to have better mechanisms for keeping cellular cooperation working, right, if you have a larger and longer lived group, which is essentially what we have with larger organisms. Other researchers have also found similar phenomena here. This is another lab that is looking at TP53, and they found that the copy number size increased as body size increased by comparing this with woolly mammoth and mastodon genomes. If we look at cetaceans, we can see that there's positive selection on a bunch of cancer-related genes in giant cetaceans. And if we want to look more broadly at variation in cancer susceptibility across species, then we can look to this huge database that myself and colleagues have been cultivating over the last decade and a half or so. We have a unbelievably large number of entries here. It's from zoos, from veterinary pathologists, and we've been able to look at the frequency of neoplasias. So those are sort of precancerous lesions, but tumors, and then also malignancy. So things that we would call cancer in many, many species. And what we've found, so this is just to kind of give you an overview of what we have in the database, but we found that there's a seemingly higher risk of cancer in mammals compared to amphibia and sauropsids. And also, and this is different actually from earlier work, that larger organisms actually have a higher susceptibility to cancer in the form of neoplasia. So these are the sort of precancerous lesions. They have a greater frequency of growths that they shouldn't have. And that kind of makes sense if you think that the process of selection inside the body, cellular competition and conflict that's going to lead to the evolution of cancer, that you're going to be more likely to have that if you have a larger organism, right? Because you have more cells that you're starting with, and that just when you have an exponential process, just having a larger starting population makes it more likely that you're going to have a problem. So this is what we see with adult weight. If we look at longer lived organisms, however, it's pretty flat. So this suggests that perhaps with longer lived organisms, maybe because they have perhaps a slower life history strategy, that there's also been more selection for cancer suppression in those organisms as well. And interestingly, with longer gestation time, there's a slight negative relationship. And again, this sort of fits with the idea that perhaps life history strategy is playing a role here. Organisms with longer gestation times, they're engaging in more parental investment, and that might also mean that they're engaging in more somatic maintenance. So they're sort of leaning on the side of investing in the future, both for themselves and for their offspring, perhaps.
Mm-hmm. I think just letting you know that we are at 25 minutes, so if you wouldn't mind wrapping it up, please.
Absolutely. So I'll just leave you with this final slide then, which perhaps not unexpected here, right? That a higher mutation rate is associated with a higher neoplasia prevalence. And so this is pretty much what you would expect if you have a higher mutation rate within the organism, then that's going to give more opportunities for selection within the organism to lead to that sort of cheating that may be compromising the organism itself. So I will just leave this up with my thank yous and be very happy to take some questions.
Thank you so much, Athena. Wow, I love this research. So Elliott, if you want to go ahead and ask the first question here, please go ahead.
I'm afraid I don't have a question to ask. I should have taken my hand down from before. Excuse me.
Let's stop sharing the screen.
All right.
Any other questions here? Comments?
I had so many questions during my talk and now everybody's quiet.
There,
I love it.
Oh, okay.
Actually, Athena, I might have a very quick question here. So I think, there's just, I want to, pinpoint the much broader theory behind the evolution of aging in cancer, really, because I think you really have to bring in, you mentioned it in the last few slides here, Medoir's curve, that the selective efficacy really decreases over age. So I think the life history components are really big in a lot of this stuff, but I know that there's also a very interesting argument. I believe it was published 2014 in PNAS by Josh Middledorf that actually cancer proliferation could potentially be an evolvability mechanism or some sort of mechanism that actually benefits populations or species, because then you'll have higher generational turnover, you'll go through more mutation and recombination events. So then you'll have higher variation in the long term. And I know he did some artificial experiments on this. So it's just a different perspective on this that I would love to get your thoughts on, actually, if you know the program. I think they're called the program theories of aging and death. Because I know it's just a different way to look at it than our old perspective.
Yeah, well, I think, when it comes to humans, that the idea of sort of programmed death, I'm not convinced that that is something that has been a strong enough force in humans, but I mean, there are other species where it's, there's obviously evidence for that, in yeast and, I mean, even within our bodies, right, having, apoptosis is, programmed cell death. But, I mean, I think that it's a topic that it would be great to sort of take more seriously in a cross-system kind of perspective, right? Because we're only really going to understand how it functions and how it's selected, and what parameters sort of allow it to exist in terms of, metapopulation structure and selective coefficients and all of that. If we have enough different kinds of systems, enough different kinds of biological, organisms and, ways that those systems work, that we can look at it in a broad context. So that's what I would say.
Yeah,
No, perfect. I know it's largely dependent on ecological conditions as well, but this is also the perfect segue into my presentation, which is next. And I apologize, Robert and Moreno, who I know who had questions. So if you wouldn't mind just putting them in the Q&A tab here, then I know then Athena can get to them here. But I am going to get to my presentation real quick here.
Well, thank you guys so much for having me. And I'm just sorry that we're not able to all be in person and go chat together after this.
Oh,
that
yes.
too. Thank you so much, Athena. It was so wonderful.
Okay. Okay, so I'm gonna swap this. Can everybody see it? Looks good?
I think so.
Okay, awesome. Well, welcome, bienvenidos. My name is Mitchell Ryan Distin. I am the co-organizer of the MLS initiative along with David Sloan Wilson, as well as a postdoctoral researcher at Prosocial World. And today I'll be talking about macro-MLS theory, so sex evolvability and major evolutionary transitions. But a lot of the forthcoming is actually in this book here, this ebook called Evolution in Space and Time. So if you want more at greater length, here. I'm going to be very general in this talk and give a lot of history. So if you want more of the specific studies, I know I have like 600 references in there, so please refer to that there. Without further ado, in this presentation, I will show how and why we need to update the causal core, the very theoretical foundations of natural selection theory, to encompass causation at multiple levels of biological organization over space and time. So traditionally, we have conceived of natural selection in a relatively static causal space, where if we have heritable variation among individuals in selective pressure, then we will invariably have selection within populations. However, we've known since at least the 1930s, and what became a scientifically validated claim since at least the 1990s, is that there's a lot more randomness and external selective influence in the processes generating heritable variation themselves, in which higher level selection can act directly on mutation or recombination modifiers, thereby increasing the causal efficacy of selection at lower levels and facilitating adaptation. And so in this presentation, I'll show precisely how and why this multi-level selective process ensues using what I consider to be the most historically validated and best empirical example of MLS, which is sex and evolvability. But to first talk about how we evolved a more complex causal core of natural selection theory over the past few decades, I need to first tell the story about how science began, how science used to be done, since the OG underlying philosophies of science, that of mechanism and causal determinism, have always acted as a significant roadblock to biologists understanding and explaining the inherent complexity of the natural world, which indirectly hid MLS theory from view for over a century. So mechanism, by the way, attempts to explain phenomena by breaking it into parts and explaining the whole thing part by part like a machine. And causal determinism is a corollary of mechanism in which scientists attempt to construct universal exceptionless laws of absolute and monocausal phenomena, usually in the form of mathematical laws or theorems that are supposed to hold constant in space and time. However, fast forward a few hundred years, and the underlying philosophies that drove science since Newton gave birth to modern evolutionary theory. So put differently, the intellectual environment that incubated early evolutionary theory, the defining scientific philosophy of the early 20th century, was known as logical positivism. So positivists such as Otto Neurath, Rudolf Carnap, Carl Hempel, etc., were particularly impressed with the logic and mathematics and technological success of late 19th century and early 20th century theoretical physics, which was back then the paragon for the other sciences to follow. They started a tradition known as the Unity of Science movement, which in its most egregious form held that all scientific terminology, laws, and theories should be reducible to physics in the long run. However, in its weaker form, the movement to unify the sciences approached scientific phenomena using an explanatory strategy known as explanatory monism or reductionism, which particularly fell under the broader philosophical umbrella of mechanism. And so these include the favoring of one type of explanation, which is explanatory monism, or one fundamental level of explanation from which others can be derived or reducible to, which is reductionism. It was often the result of emergent phenomena being reducible to lower level events. Thus, early biological methodology was fashioned after physics because that's just how everybody thought we should perform science. Before the modern synthesis around 1918, 1956, biology was seen as a second science. There were many competing theories of adaptation, not just natural selection, but also orthogenetic theory and Lamarckism. And all the subdisciplines of biology, such as paleontology, genetics, physiology, were largely divided. However, after the modern synthesis, evolutionary biology and biology in general became seen as respectable scientific disciplines. There was universal acceptance of natural selection as the primary theory of adaptation, and biological knowledge became integrated into coherent theoretical framework, with evolutionary theory lying at the epicenter of biology. So what caused all these significant changes during this era? Well, primarily the adoption of a robust mathematical program that quantified evolution, otherwise known as population genetics, lent evolutionary biology a new legitimacy within the broader intellectual zeitgeist of logical positivism. So a new biological methodology was to be fashioned after the acceptable sciences of Newtonian physics and chemistry, while also allowing biology to preserve its autonomy from these sciences. So all three of the founders of population genetics, R.A. Fisher, C.O. Wright, J.B.S. Haldane, were attempting to bring biology on par with the physical sciences and drew explicit exemplars from these exact and rigorous sciences. And this was how the population geneticists modeled evolutionary change at a population level, with the four causal forces of selection, mutation, migration, and drift acting on individuals within a population to cause evolutionary change. And this is how I was originally taught evolution at university, and many still teach it today. So yet the overarching reason why we as a discipline have persistently failed to produce better theory, the overarching reason why population genetics has largely failed to produce better, more externally valid theory, is because we cannot use the same explanatory strategies as physics due to the messy causation inherent in every biological system. So most biological phenomena are variable in space and time, and the causal effects of biological traits are nearly always multifactorial. They ensue at multiple levels of biological organization with upwards and downwards causation. And they all come with a complex evolutionary back history that was likely instantiated by every evolutionary process, that of selection, variation, drift. So add on to this the fact that organisms can influence their environment and vice versa, necessitating a reciprocal model of causation. Yet most importantly, we also have to consider the capriciousness of ecological variables. Thus, the messiness of biological causation is precisely the reason why we cannot fashion biological methodology after physics, why we cannot use the same theoretical and explanatory strategies to explain our causally complex phenomena. Which leads us to the fundamental idea of evolution, the idea of change. Evolution is about biological change over time. It is a surprising historical fact that for the majority of evolutionary research, we have rarely studied how evolution typically unfolds in nature and changing ecological environments over space and time. So this is the primary insight behind what I see as a second major biological synthesis, what I refer to as the second synthesis. So I think we are undergoing something of an empirical revolution in biology, where we are beginning to place more of a philosophical emphasis on the ecological validity of our empirical studies. So I can't go into all the fun and necessary history here, but just note that while ecology did not play a major role in the early development of evolutionary theory through the vehicle of population genetics, it did play a much larger role in the overall acceptance of the population genetic viewpoint of evolution during the modern synthesis, largely due to the background efforts of early evolutionary ecologists such as Dobzhansky and E.B. Ford. So early evolutionary theory was thus particularly influential on early evolutionary thinking, but it did not begin to inform evolutionary theory until about the 1980s, which is when we begin to see an explosion of field studies of natural selection, largely due to the methodological advances spurred by establishing a quantitative standard to study natural selection dynamics in the wild, known as the Landian-Arnold framework. So as reviewed in John Emler's Natural Selection in the Wild, we began to finally understand how evolutionary dynamics play out over longer timescales and frequent spatial changes, which is furthered by the establishment of the Evolutionary Rescue and Eco-Evolutionary Dynamics Research Fund. Thus, from the 1980s onwards, I think an entirely different, novel, and emergent picture of evolution has come to the fore. So putting this all together, this is why I see recent developments in evolutionary biology and ecology as another whole-scale biological synthesis, because its underlying causes and consequences are similar to that seen in the first synthesis. So before the second synthesis, we generally thought of evolution as a slow and gradual process. Natural selection dynamics as they play out in the complex space and time of nature were relatively unknown, and the causal models or theories we constructed to explain evolution and natural selection were therefore largely abstract and reductive, because they had little empirical input from ecology. Now, following the subtle and organic synthesis between ecology and evolutionary biology over the past 40 years, we now know that evolution can proceed at faster rates and thus on similar timescales to ecology. We have seen an exponential increase in our knowledge of natural selection dynamics, expounded in all the recent reviews of field studies over the past 20 years. Which brings me to my last point here in red, which is more prescriptive than descriptive. So without question, we have recently experienced a massive increase in our knowledge of evolutionary and selective dynamics due to the second synthesis. And whenever we see such a massive increase in our epistemic reservoirs in the sciences, we should meet such progress with theoretical reflection. So I therefore think we are existing in an intermediate stage of scientific progress that warrants turning all the new knowledge revealed by the second synthesis into useful and predictive theory, into externally valid theory. So this is where the philosophy of biology can be of great use, but let's put this idea on hold for the time being and get into the evidential background for everything discussed herein. So what I've referred to as the paradox of adaptive genetic variation is the longest standing and most severe explanatory problem in the history of evolutionary biology, because beginning with Darwin, evolutionary biologists have routinely failed to explain the evolution of adaptive genetic variation mechanisms of sexual recombination, mutation, horizontal gene transfer, genetic transposition, and whole genome duplication events. And this problem is still an active one today, which is why it is such a useful example to use to demonstrate all the known issues with evolutionary theory, because its severity shines a particular spotlight on the philosophical anachronisms that still pervade in evolutionary theory that we originally derived from physics. So for a quick review, again, this slide shows the basic modern synthetic model of natural selection, or how we used to think the process of natural selection played out. Essentially, the modern synthetic model saw natural selection as a two-step causal process that ensues in a linear one-way flow of causation from the first step to the second step. So in the first process, you have mutation and recombination mechanisms causing variation, but it is important to note that they cause variation randomly, meaning that they aren't generally influenced by the external selective environment, or that their subsequent phenotypic effects cannot be predicted, which is just fancy talk to say that mutations aren't directed, which I believe this latter sense of randomness has actually held up in the literature today, but not the former. So the second step is what we classically understand as natural selection, which is when an environment selects for the variants that are the best adapted, i.e. have the highest fitness, while leaving or purifying less adapted variants. But of course, as I previously mentioned, these two causal processes don't ensue independently of one another in space and time. Natural selection can act on mutation modifiers, thus enhancing its own causal efficacy to cause adaptation, or a selection for selection, let's say. But what's also wrong with this model is the implicit assumption that was tagged on from very early on, that natural selection only causes adaptation at lower levels of biological organization, at the genetic level of selfish genes, or at the individual organismal level, most prominently because our methodologies were limited to within population analyses for most of evolutionary research in the 20th century. So this assumption was born out of the intellectual context that Stephen Jay Gould referred to as the hardening of Darwinism, which is when reputable evolutionary biologists began to infer from their mostly statistical interpretations that natural selection only acts at lower levels of biological organization at the behest of lower level entities such as selfish genes or individuals. And this whole debacle, known as the group selection controversy of the 1960s, started when the ecologist Wynne Edwards posited that individuals may sacrifice their reproductive interests for population control, which was a naive group selection type of claim. And so such a provocation moved several of the most reputable evolutionary theorists to specifically attack the idea of group selection, which led to an overall suspicion of all higher level and emergent phenomena in evolutionary biology. So following this period, natural selection was universally seen to operate at lower levels, procuring adaptations that benefit selfish genes or individuals within a population. And as you can see, this idea continues through to this day, best exemplified by this quote by Jerry Coyne. As evolution predicts, we never see adaptations that benefit the species at the expense of the individual. Thus, some of our best and brightest within evolutionary biology today still cling to this assumption. Yet quite interestingly, and this is what we were talking about before here, Robert, is that the only exception to the individual selection assumption always came from sex. So sex was the only phenomena that even the strictest of reductive Darwinians, such as R.A. Fisher, G.C. Williams, Maynard Smith, ever considered to not work within their theoretical models of natural selection. So in my book, I go into the interesting back and forth between Maynard Smith and Williams, who first sought to explain sex within their reductive purview. However, after repeated failed attempts, Williams and Maynard Smith finally conceded to the idea of higher level selection as the primary causal force underlying sex and its maintenance. So here is a quote by Graham Bell that perfectly sums up this situation, this historical situation. So it was the very success of this attack on group selection which led population biologists to realize how embarrassing sex is. So most supposedly altruistic behaviors were quickly found either to have concealed advantages for the individual, which we now know is also not true, or else to be directed towards the welfare of closely related individuals. But sex appeared to fit into neither of these categories. If it permits the rapid mobilization of genetic variation, then this may be a matter of vital concern for the population, but does not in itself concern the individual. So evolutionary biologists thus found themselves on the horns of a dilemma. Either the apparently unsatisfactory hypothesis of group selection was indeed an adequate explanation for the maintenance of sexuality, or else quite a different hypothesis framed in terms of natural selection must be sought. So as exemplified by Bell here, the unrelenting commitment of many 20th century evolutionary biologists to a reductive model of natural selection caused many of the same architects of this reductive theory to notice an implicit and profound theoretical incongruency between natural selection theory and the available evidence on sex. So the paradox of sex was thus officially recognized in the 1970s after reductive causal interpretations of natural selection became entrenched in evolutionary theory following the group selection controversy, thus raising the origin and maintenance of sex as a major explanatory issue for evolutionary biologists that largely continues to this day. So sex is still widely regarded as one of the major unexplained phenomena in evolutionary biology, what Graham Bell refers to as the queen of problems in evolutionary biology, mostly because it is incredibly pervasive in eukaryotes. So why does sex present such a paradox to reductive theory? Well, because sex is well known as an emergent and species level feature that is also extremely costly to the individual. It takes time and energy to find a mate. Individuals only leave 50% of their alleles to offspring, and there's a significant evolutionary detriment to having the amount of reproductive individuals in a species, otherwise known as the cost of males. So why do we sex then, given all of its cost at the individual level? Well, sex does one thing in particular, and we have actually known this since August Weismann in the late 19th century, that the genetic mixing seen in meiotic recombination is known to accelerate and facilitate adaptation due to the novel genetic variation that it produces at the population or species level. So empirical studies have repeatedly found a strong evolutionary benefit that comes with outcrossing or the interbreeding of unrelated individuals, as opposed to self-fertilization, because of the evolvability benefit that it confers to higher level entities. What is less recognized today is that adaptive mutation functions as the exact same thing as sex for much the same reasons, coming along with the exact same explanatory issues. So beginning in the 1970s and 80s, researchers began observing increased mutation rates when they placed bacterial populations under stress. And so this began to erode the fanciful modern synthetic notion that all mutations are random, and began to imply that selection may actually increase mutation rates when a population is stressed, despite deleterious effects to individuals within the population. So the evolutionary biology community has undergone something of a bifurcation in models of mutational dynamics over the past 20 years. With a non-adaptive models of mutation, such as Drake's model or Lynch's drip barrier hypothesis, struggling to maintain the past reductive theoretical tenets of natural selection, seeing natural selection as only a force that decreases mutation rates to conserve individual offspring viability. However, a new model has slowly and subtly been building in the literature known as the adaptive models of mutation that argues that natural selection can act to increase mutation rates so as to increasing the probability of fixating an advantageous genetic variant and facilitating adaptation when a population is stressed or in a novel environment. And so I'm not going to go into a great detail on all the evidence that now underpins the paradox of sex, and why we need a multi-level selective theory to explain this evidence. So if you want more, please refer to my book, but just note that a lot of this research stems from ecological studies and studies that look at the long-term evolutionary dynamics and the evolutionary rescue of populations. And that's a key point here. And same with the adaptive mutation literature. Note the connection between adaptive mutation and the evolutionary rescue of populations. Thus, the big idea here is that adaptive genetic variation mechanisms are emergent adaptations that benefit higher level entities when placed in novel, stressful, or capricious environment, therefore driving adaptation at lower levels. So this is not a new idea in the literature by any means. So this is how most evolutionary ecologists and conservational biologists think most species respond to environmental changes, from demographic rescue, phenotypic plasticity, or developmental mechanisms, or genetic adaptation, which is itself divided into these three processes. And these three elements are what I refer to as genetic evolvability. And if you want more on how I'm conceiving of genetic evolvability, please refer to my chapter in this recent book. And so a recent meta-analysis of 19 long-term studies performed in the wild, which is a crucial part here, also published in Science here, found a 2 to 4 times larger additive genetic variance in relative fitness, implying that genetic adaptation is a much more significant causal factor in the rescue of species than we previously thought. So most natural populations harbor an extensive amount of genetic variation. So why is this variation there? It doesn't benefit individuals, at least not in the short term. It doesn't benefit selfish genes. It's there because natural selection truly has foresight because of what has happened in hindsight. So environmental unpredictability is the only predictability in evolution, hence why most species harbor an extensive amount of genetic variation. So in light of all the recent evidence on evolvability, we need to pluralize natural selection theory so that it's not strictly reducible to causation at lower levels, but is capable of explaining adaptation at higher levels and over longer timescales. So we need a pluralistic model of natural selection that really takes into consideration the idea of multi-level selection to explain complex, selective, and long-term dynamics. And so why do we need to do so? Because I believe it leads to more virtuous theory. It increases the explanatory depth, the theoretical coherency, evidential accuracy of our theoretical mainframes. And this is, by the way, precisely what science needs, a way of comparing and contrasting competing theoretical models using these virtues. So thus, to explain sex and evolvability, we truly need a multi-level selective framework to explain how selection operates between various and conflicting levels of biological organization, or how lower level features such as stress-induced mutagenesis or recombination affect the survival of populations or species over time, because evolvability is typically a population or species level trait. So I don't know if anybody here is a Twilight fan, but if you are, you'll get this probably better than everybody else. But what this research shows is how evolvability operates within a multi-level selective framework. Since stable environments typically allow individual level selection to dominate, but when environments change, higher level selection for evolvability becomes the major adaptive interest. And so this is precisely how evolvability actually operates in nature as a higher level fitness determinant, meaning that natural selection does not only select for greater individual fitness measures and higher fitness peaks, but also for the flexibility to traverse the adaptive landscape and reach new fitness peaks. So in my book, I have attempted to place evolutionary theory within its historical and philosophical context, thus revealing the philosophical anachronisms that I believe are presently weighing down biologists and impeding scientific progress. So the main issue I've noticed is that we have historically not looked at evolution in space and time. We have conceived of evolution as a relatively static process that happens in relatively static environments. Our methodologies only revealed short-term and individual level evolutionary phenomena, thus making our theory particularly reductive and not very externally valid. And now over the past 40 years, evolutionary biology has undergone something of an empirical revolution, whereby we now look at evolutionary and selective dynamics in the long term and over frequent spatial changes. And as such, I believe we're beginning to reveal long-term evolutionary phenomena and long-term selective phenomena, such as evolvability and multi-level selection. And so putting it all together into one graph here is just a general schema of what I believe is the literature is showing. Is that one here is this is how we have traditionally looked at evolution from the perspective of within populations and at shorter timescales in relatively stable environments. So in periods such as these, natural selection will tend to place an adaptive emphasis at the lowest levels of individuals or selfish genes, thereby selecting for enhanced individual fitness metrics. Evolvability and other long-term phenomena such as multi-level selection are either dormant due to low environmental variability or appear to be dormant merely due to our timescale choice. So I believe here at number 2, an increase in environmental variability causes adaptive radiations to ensue and natural selection to select for higher level features. During these periods, the evolution of weird evolutionary strategies such as social cooperation, the formation of new levels of biological organization and or evolvability becomes more likely. So adaptive interest will tend to shift to the population and or species, meaning that natural selection will tend to favor higher levels of biological organization and select for higher level fitness determinants, such as evolvability. So during these large scale changes in environmental conditions, when environmental variability and therefore selective pressures are at its highest peak, such as mass extinction events, we see the evolution of the weirdest evolutionary strategies, such as whole genome duplication events, which are typically an evolutionary dead end in stable conditions. But this is also when we are the likeliest to observe cooperation evolving between completely unrelated species or the emergence of entirely new levels of biological organization, i.e., major evolutionary transitions. And finally, following more environmental changes and over time going through all these little hills, these ebbs and flows, I believe the traits become honed in what initially started as interlevel conflicts from the evolution of new adaptations at the new levels are actually finally resolved over time. And I believe this is what's happened in the sex literature, because we've seen that sex enhances both robustness but evolvability at the same moment. So thus coming full circle here, since the days of Newton, scientists have focused on mechanism as our defining scientific philosophy underlying everything that we consider as science, which relatedly meant that we emphasize finding monocausal or absolute phenomena, i.e. laws. It also meant that we screened off interferers, we modeled causes in simulations and or simplified assumptions, we isolated test subjects in constant laboratory conditions, and we experimented with similar or identical test subjects, and we also constructed causal models that only apply ceteris paribus. So, however, I believe the general idea of the second synthesis is that we are now finally becoming a more mature scientific discipline with our own philosophies of science, what is becoming the philosophy of biology, that help us better explain multifaceted and complex phenomena within our own domain. So while the first synthesis legitimized biology as a respectable scientific discipline, the second synthesis autonomized biology and gave us our own philosophy. Thus, we now need to form multi-causal, dynamic causal models that are capable of explaining ecological complexity. I believe we need to model causation at multiple levels. We need to experiment with multiple varying species, not just model species. We need to enhance the ecological validity of our laboratory experiments to make them more lifelike, more representative to natural environments. And we also need to embrace the philosophies of integrative pluralism, emergence, in systems thinking. And I believe if we do all these things, I really do believe that it'll lead to just a better biology in general here. So without that, that is the end of my presentation here. Yes, 25, I got it right on the dot. I was very worried about that.
Okay. If nothing else, this symposium is revealing the big picture. And now we have hands raised. So Jeremy.
Yes, good.
Hi, Mitchell, thanks. That was a really interesting talk. Lots of really good stuff there. I just wanted to note, though, that for like mutation rate and recombination rate, that those models don't require any kind of multi-level selection or selection at the group level. So I think there's definitely, there's some conceptual issues there that maybe need working out a little bit.
So I may push back on this. So I believe it's there, but it's just not explicit. I know,
but there's there's infinite population size. There's no population structure. The only thing that's different in those models is that you've made the environment variable. I mean, these are sort of very classic modifier, multi-locus, infinite population size models.
Well, all I could say to that point is refer to Sally Otto's work, because I think she talks about this in greater depth and breadth than anybody, and talking about how depending on the ecological conditions and how well adapted a species is to their ecological conditions, they will either favor, haploidy or individual level selection, or group selection in diploidy. So yeah, I think she gave a brilliant lecture on this in 2017 as the presidential address of the evolution meeting, actually. So actually, let's talk more on that, Jeremy, because I have a lot more to say on that.
No problem.
Yeah, all these to be continued. We're encouraging people to join the multi-level selection initiative precisely to work through issues like this.
Mm-hmm. So Vincent, if you wouldn't mind.
Hi, Mitchell. Thanks. I want to congratulate you on the clarity of that talk. That was really great. My question is, your presentation of the history and the philosophy of multi-level selection got me thinking, have you thought about or found any explicit kind of or subtle connections to eugenic thinking and the development of capitalism and the change towards kinds of Marxist thinking and sociobiology and that sort of thing? So I've been reading Lewontin's Dialectical Biologist. It's really got me thinking about how in multi-level selection, we're thinking a lot about altruism and groups, right? And in individual selection, we're not. We're thinking about kind of tooth and claw. And so have you seen that? And is that something that comes through when you're doing?
Oh, no, certainly. I think that this will be, better explained in Stephen and David's talk, the sociopolitical dimensions of multi-level selection theory. But I think that has always been a constraint, bringing in the sociopolitical history to really belief in group selection. And I believe the reluctance of a lot of individual level selectionists really comes from that fundamental tension of 20th century sociopolitical thought between individualism and collectivism. So I think no doubt about it that plays a big, and I know it played a big role actually in Dobzhansky's thinking. So he was more on the collective end of this, and he he was a big early progenitor of evolvability and multi-level kind of thinking. So yeah, definitely a big broader history that actually nobody's done significant work on. So Vincent, I also implore you to do that if you really want to, but also please join our initiative, because this is actually what we're about to talk to make a main emphasis in the MLS initiative, expanding MLS theory past just biological and genetic applications. So
yeah, let's see if Robert can sneak in a question. I'll withhold my own hand so we can keep on track.
Yeah, it's a comment, really. If someone else has a question, I'll hold off.
No, go ahead.
Yeah, so you use phrases like Darwinism hardened and the modern synthesis failed. And I get the sense when I hear phrases like that, it seems, and please let me qualify this by thanking you for bringing up this really cool stuff, which I love talking about. But it sounds like a bit of a political program more than anything. And I worry that some of the really great things that someone like George William did in the 60s, I mean, he was right that male lions don't engage in infanticide because they anticipate future resource scarcity. And there are, you know, most of what we're passing judgment on in terms of the overkill in the anti-group selection movement of the 60s and 70s, we're all doing that in hindsight. And I think surely we still owe a big debt to people like Dawkins and Maynard Smith and George Williams for even in terms of sex ratios. I mean, they usually are close to 0.5, as Elliott pointed out earlier. He missed some important cases which it isn't. So I don't want you to get the impression that I'm a diehard individual selectionist or anything. I'm a really big fan of this whole thing. But I do worry when I hear phrases like, you know, Darwinism hardened and the modern synthesis failed, because we're all standing on the shoulders of these people, and the dismissing individual adaptation or individual selectionism is no better than dismissing group selection like you know, Williams would have done. So that's my comment.
No, love it.
Yeah,
I'm going to respond to that in the chat here because we really have to get to Cesar's talk. So, Cesar, if you wouldn't mind introducing yourself and sharing your screen.
Do you see my screen?
Yeah.
Okay. So hello everyone. I'm César Marín. I live in Chile. I'm originally from Colombia. And during my bachelor, I was very interested in multi-level selection and also during my PhD. But as it always happens with this kind of topics, I had no mentor, no PhD advisor that knew about the subject. So I started to work in something completely different, which is mycorrhizal ecology, and I am a mycorrhizal ecologist by formation. And over the last 2 years, I will say, I finally started to combine both subjects, both topics, mycorrhizal ecology and multi-level selection. And this is just the beginning of that. So my talk is multi-level or functional selection in mycorrhizal symbiosis. First, I want to start by saying that mycorrhizas are a big deal. Then that the mycorrhizal mutualism is context dependent. Then a few slides on units of selection in mycorrhizal symbiosis, because I think that defining the specific units of selection will solve many of the discussions and debates, not just in mycorrhizal systems, but in any system. And then our last proposal, which is a functional team selection. So very briefly, an introduction about mycorrhizas. Well, of course, this is a symbiosis between plant roots and fungi. There is a direct exchange between photosynthesis products and nutrients and water, and protection against biotic and abiotic stress tolerance, represented here in this nice figure from a paper of this year. Then they are, of course, very important. They are present in 86% of land plants, according to the last estimates. Most of the plants that we know, most of the major crops associate with mycorrhizal fungi. The ones that do not associate with mycorrhizal fungi are plants like carnivores or plants living at very high altitudes. But most of plants associate with these fungi. And in evolutionary terms or in geological terms, they, sorry that my throat is a little hurt. They evolve concurrently, they evolve at the same time with land plants. So actually, the first fossils of mycorrhizal fungi appear before the first root fossils of land plants. And if you see here in the slide, the oldest known spore of arbuscular mycorrhizal fungi is very similar to the modern day spores. And they deeply control or they have a very strong effect on global biogeochemical cycles. So the people from ecosystem ecology have studied this a lot over the last 15 years. They have studied how different mycorrhizal types, for example, affect carbon storage, or how they directly affect the nutrient economy of plants. as shown here in this study from last year, where the authors analyze 100 and almost 200 data sets and show that the amount of carbon that they store, that the mycorrhizal hyphae stores, is huge. It's equivalent to the 36% of the annual CO2 from fossil fuels, at least temporarily. So there is no doubt that they are hugely important, and they stabilize the soil organic matter. Then one of the most important messages of this talk that I wish that people have very clear is that, at least in mycorrhizal context, I'm not talking about the other types of symbiosis, but at least in mycorrhizal context, talking about about symbiosis, sorry, is different of talking about mutualism. So mycorrhizas exist across a mutualism to parasitism continuum, which this concept is very ecological, but if you think about it, is also a very multi-level selection topic, or a very multi-level selection concept. So this was proposed many years ago, 26 years ago, and at the beginning there was a lot of pushback against this concept, but now it's more accepted. When you look at into a root, you see two types of structures: the arbuscules, where the nutrient exchange occurs, and the vesicles. So vesicles is basically where the fungi stores the nutrients for itself, and is not transferred to the plant. So in this study from more than 30 years ago, Nancy Collins Johnson, she showed that fungi growing in fertilized soils, they produce way more vesicles, and they colonize less the roots. And in soils without fertilization, the opposite happens. So in soils with few nutrients, with a low concentration, particularly of phosphorus, they produce a lot of arbuscules. So it means that they are sharing a lot of nutrients. So this was like the beginning of this concept. Then, of course, this has been meta-analyzed. And again, the meta-analysis show that under luxury conditions, under soils with a lot of phosphorus or a lot of nitrogen, the plants with mycorrhizas, the plants inoculated with mycorrhizal fungi, actually grow less than the control which is not inoculated. And this at the beginning was very counterintuitive, but again, a lot of evidence on this. This study in this Ecology Letters article, all the 130 studies included there are in pots, because of course you need to have a control, and a sterile control. But then when people started to do this in situ, as this study from last year in Switzerland, they inoculated 54 corn fields across northern Switzerland. And again, a very interesting result. They found that only in 25% of the cornfields, the plants with mycorrhizas grow more. Then in 50%, there are no differences. And then in 25% of the fields, the inoculated plants grow less. And of course, they measure about 53 environmental and biotic variables, including the microbiome of the soil before inoculation. And they run nice statistical model, and they found that the variable that predicts the positive results is soils with a high initial fungal pathogen abundance. And this has not been found before in the literature, but again, because most of the studies are conducted in greenhouses, but this is very appealing, a very appealing result. Then a series of studies starting in 2010 by Nancy Johnson from Northern Arizona University and many other people, they found this very interesting local adaptation between three components, between the plants, between the mycorrhizal fungi, and between the whole soil microbiome. So when these three components are sympatric, when they come from the same site, these plants withstand better. They withstand better drier conditions or hotter conditions than when one of the components is allopathic. So this is like a clear evidence that there is sympatric adaptation in these three components. And this is another context in which the mutualism is enhanced. And then to add a little bit more complexity to the system, since 2016, some people are studying the component that we call the hyphosphere. Similarly, as plants have a rhizosphere, which is basically the soil right next to the roots, the mycorrhizal hyphae, the extraradical hyphae, the hyphae that lives outside the roots, also has an hyphosphere and a very diverse microbiome in those hyphae. And what people is finding is that this bacteria that lives in the hyphosphere is able to mineralize phosphorus, which is crucially important, particularly for arbuscular mycorrhizal fungi. Always you read in the literature that arbuscular mycorrhizal fungi is associated with phosphorus nutrition, but they actually don't degrade the phosphorus. So it was always kind of a mystery on how actually the biochemical process or the biochemical process was occurring. And what these Chinese researchers found was that is the bacteria living in the hyphae, and that there is a core microbiome. And here is important to notice that this core microbiome is not the same taxa, but the same functional guilds across very different systems in China, and also in a microcosm experiment. And please remember this, because this is important for a definition of the holobiome that I will give later on. Here is a nice interview to the authors of this article and the implications that this process has. And then there was this manipulative experiment also from last year, different authors, but they show basically the same. They separated the bacteria which belong to the genus Streptomyces. They separated the mycorrhizal fungi and the plants, and they made all the three combinations. And basically what they found is what I highlight there in the conclusions, that arbuscular mycorrhizal fungi do not function in isolation, and that the plant mycorrhizal symbiont can recruit beneficial bacteria that supports the symbiosis. In other words, mycorrhizal fungi deeply depends on this bacteria living in the hyphae. And to complicate things even further, in mycorrhizas, we knew for a while, for more than 20 years, that there are bacteria living inside the spores, but it was kind of a forgotten subject. And recently, this team in Ireland, they bring back this subject, studying the bacteria living inside the spores. And they found that one spore could have more than 200 bacterial species. What are they doing inside the spores? Basically, we don't know. There is not enough information to say what they are doing. But they suggest that they could be also conditional mutualists. So mycorrhizal mutualism is context dependent. It depends on soil nutrients and is enhanced when nutrients are limited. It depends on the rest of the soil microbiome, and it's enhanced with a high initial pathogen abundance. In other words, mycorrhizal mutualism is enhanced under stressful conditions. It's also enhanced under sympatric conditions, where the soil microbiome and the mycorrhizal fungi and the plants are from the same site. And I don't show this here in the slide, but the people from restoration ecology, they know this pretty well because several meta-analyses show that whole soil microbiome restoration or inoculation is one of the only few techniques that always works, and especially if the soil microbiome comes from the same site. And lastly, it's also clear that mycorrhizal mutualism depends on this iosphere, on this microbiome living in the IFI. Then I think that is with this picture, I think that is necessary to think about units of selection in mycorrhizal symbiosis. So Javier Suarez and Lisa Joy last year published this very nice Cambridge Elements book on which I wrote a review in Evolution. And well, the subject is not new. Lisa has been writing about this for decades, but it's very important to keep in mind the different units of selection of the different three meanings. And for me, the most important thing is to know that they don't need to occur at the same level of selection. So we need to differentiate between levels and units of selection. And in some cases, like maybe in major evolutionary transitions, it's possible that a single entity has the three roles, that a single entity is an interactor, a replicator, and a manifestor of adaptation. But this doesn't have to be the case always. Most commonly, you would think about organisms as interactors and then genes as replicators. But what I am arguing for the field of mycorrhizal ecology and microevolution is that we really need to think about this. This slide shows kind of some models, some proposals, and some empirical evidence that show, for example, natural selection occurring at the nuclei level, because one spore of arbuscular mycorrhizal fungi can have hundreds of nuclei. We propose that there is selection going on at the iosphere level or at the holobiome level. Some people have shown that whole groups of spores, meaning population level, also are present phenotypic variation and present differential fitness, which will make them an interactor. So what I argue is that we really need to think about this in this specific hierarchical view that the mycorrhizal systems have. You see here some examples of that. The spores can have dikaryon and homokaryon nuclei. And this group in Amsterdam, they are proposing that indeed some nuclei have a selective advantage over others. And if you see this photo, there is no segmentation. So this is a single cell, which is amazing. And the nuclei basically travel across this huge cell. And regarding the fungi living outside the root, they also, under certain conditions, form these huge sporocarps, which is not usually how you see mycorrhiza in nature. You usually need a microscope, but in these cases, you can see them to the naked eye, and we really don't know how or why this aggregation of cells is occurring, of spores is occurring, but definitely it seems like they at least will constitute an interactor if we stick to the Lisa and Javier's definitions of units of selection. So finally, we are proposing the idea of functional team selection, which after seeing Joan's presentation earlier this morning, is very similar to the host-mediated microbiome engineer. Is kind of has the same elements. First, in this brief commentary, we suggest or define a mycorrhizal collective as a plant host and some mycorrhizal fungal traits and some traits of the ionosphere. In the example that I gave you, for example, it can be any host and mycorrhizal traits related to stress tolerance, for example, and trace of the ionosphere, particularly phosphorus mineralization or phosphorus degradation. And this is very similar to the stability of traits concept of the holobiome by Javier Suarez from 2020. But to be honest, we didn't knew this concept when we wrote this. So for me, it's always interesting to see this, that people from many different fields come to similar concepts. And this, of course, will depend on nutrient context. So by functional team selection, we mean that that plant holobiomes, although we don't use the word there because there is also a microbiome living in the leaves of plants, and which is called the phyllosphere. So maybe the plant holobiome concept should also include this phyllosphere. But anyway, we say that plants can recognize and cultivate beneficial microbes, and this includes mycorrhizal fungi, and this includes also a cultivation process of the mycorrhizal fungi towards the ionosphere. Then these plants and microbiomes are basically complex adaptive systems, and plants' adaptations are emergent properties of these complex adaptive systems. But this is context dependent, and this can change over the lifespan of a plant. For example, with the plants that I work, native conifers from southern Chile, they can live thousands of years. So of course, we need to take into account the huge difference into the lifespans of plants and these microbiomes. But anyway, the function of these plant microbiomes is context dependent. And these rhizosphere microbiomes are especially and temporally dynamic, which we think that most current models and definitions of holobiomes maybe don't take into account this temporal and spatial variability of these microbiomes. This is a graphical representation of this. On limited soil resources, is easier that plants select what we call winning arbuscular mycorrhizal fungi and winning microbiome, in contrast with luxury conditions where this process do not happen. And there is a lot of evidence of that. Recently, I published an article about the arbuscular mycorrhizal fungi of vineyards in Chile, and especially in the older vineyards with 500 years, and a lot of nutrients in the soil, a lot of conventional fertilization. You almost find no arbuscular mycorrhizal fungi, 1 or 2 species, and they are probably doing nothing in that. So that would be an example of a luxury soil resource. And this, of course, gave us a space to make some agronomic recommendations, basically to starve a little bit the plants, but also to add a minimum of initial resources for the fungi. Same with water. No tillage, of course, is a huge always recommendation for sustaining these networks in the soil. And as conclusions, I want to emphasize that soil nutrient and microbiome context determine how mutualist or not is this symbiosis. As a second conclusion, that there is sympatric or local adaptation between plant hosts, arbuscular mycorrhizal fungi, and soil microbiome. The third one is that the iphosphere, this microbiome in the IFE is crucial for the functioning of the symbiosis. And the most important is that we need to understand the different units of selection across this hierarchy of mycorrhizal symbiosis, from nuclei to groups of plant holobions. And with that, thank you.
That's so awesome. We've been just nailing it with respect to our 25 minutes here. So great. So who has questions? I feel that César, I mean, I love the whole thing, but the emphasis that this could go in the direction of parasitism or mutualism, and that selection is truly taking place at multiple levels, is exactly what one would expect. And I think what makes it so interesting and so imperative, I think all the talks, is not that higher level selection always takes place, not at all. Often special conditions are met, very special conditions are met, but that the only way to find out is on a case-by-case basis. And the one thing that cannot take place is that for higher levels of selection to be discounted just in any kind of categorical way, I think. And especially the work of what Michael reported in the lab and what you reported in the field. It's just like variation is never in short supply, and that's what you expect from complex systems. There's no scale at which phenotypic variation does not exist. It's not the case that you end up such a large scale that you get that sampling error curve that never happens. And this is true for cultural evolution also. There's no scale at which there's not cultural variation. National scale, it's ample variation, millions and billions of people. So I think that if you look at the three ingredients of selection, variation, selection, and heritability, the variation is never in short supply. And the heritability does exist at higher levels, as Michael has shown. Then it's a question of patterns of selection, which are often environmental. Just basically the differentials are operating on collectives. And so none of this can be categorically discounted. And so what that requires is the hard work of studying the entire metasystem and all its components, which is a tough assignment. And I think that actually segues to our next talk, which is on animal behavior. I know the animal behaviorists in our groups have been saying, gee, it's tough to have to do this kind of research because it just means, typically, if you're an animal behaviorist, you're like studying one population. But the theory calls upon you to expand your scope and to study metapopulation. You can't, and there's no alternative to that. You just have to do that. There's no other theory which makes that unnecessary. So anyhow, boy, I'm glad I got that off my chest. So
No, yeah, definitely. In mycorrhizal ecology, the mutualism to parasitism, for me, is another way of saying multi-level selection. Basically, is different wording, but it's the same process. I forgot to mention as well that regarding the how new mycorrhizae colonize plants, this is a reconstitution process, but the endobacteria that I show, they are vertically transmitted. So it's different kinds of inheritance going on at the same time in the same systems.
Yeah. Elliott, did you have a?
Sorry, I don't.
I think we have to move on to Robin here. So Robin, if you wouldn't mind introducing yourself, and yeah, perfect segue into your talk here.
Sorry, I forgot to unmute myself. Give me one moment just to get everything set. No, wait, 1 second. Get everything set up. Okay. How can I move this? Okay. So you all should just be seeing my screen now, is that correct? The slides? We good? Okay, great. So yeah, that was the perfect segue. Thank you, David, for just setting up this talk so well, to talk about the importance of looking at multi-level selection empirically and also the hard work of doing it. So I just wanted to start by thanking both David and Mitch for inviting me to speak to for organizing and inviting me to speak at this special event on multi-level selection theory. So my name is Robin Costello. I am currently a postdoctoral researcher at Auburn University, but I'll be starting as a faculty at the University of Buffalo this fall, which I'm really excited about. And today I'll be sharing the work I did for my dissertation, which measured multi-level selection in experimental populations of forked fungus beetles, and which adds to this empirical evidence of multi-level selection. So I did this dissertation work at Mountain Lake Biological Station in Southwest Virginia under the guidance of Butch Brody and Vince Formica, and with some support from Phoebe Cook. And as we've heard throughout this special event, both individual and group phenotypes influence individual fitness. Most estimates of selection calculate the relationship between an individual's phenotype and their fitness. However, group phenotypes, so as one example, the sex ratio of the group, can additionally influence individual fitness. And when both individual and group phenotypes simultaneously influence individual fitness, this is when multi-level selection occurs. And I just want to note, especially given the wonderful talks we've seen about the history of multi-level selection, that multi-level selection has been understood as the effect of group phenotype on group fitness along the lines of the good of the group. But here I'm considering the effect of group phenotypes on individual fitness. And that's an important distinction. And a growing body of literature is demonstrating that both these individual and group traits can influence individual fitness. Yet multi-level selection is rarely quantified on social behaviors. And this is surprising because social behaviors inherently involve interactions among individuals that generate group phenotypes. And in many cases, these group phenotypes that are generated from individual social behaviors are emergent behavioral properties of groups. So they're more than just averages of individual behaviors, and they can't be described at the individual level. So some examples of these emergent group properties include schooling in fish, or nest building in bees, or pack hunting in wolves. And so with this potential for both individual and group features of social behaviors to impact variation in individual fitness, multi-level selection may be a really powerful and also understudied force that shapes the evolution of social behaviors. And social networks provide us a unique opportunity to measure multi-level selection on these social behaviors. And that's because social networks describe both individual and group characteristics of social interactions. So to illustrate a social network, let's just take this group of elephants, and we can represent each individual elephant with a node and connect these nodes with lines whenever social interaction between two individual elephants occurs. And so then as we monitor this group of elephants, we can build a social network that maps all the social interactions and these multi-level social interactions that occur within this population. And so here we can see this social network, the width of these lines represents the number of social interactions that occur between any two elephants. And so because social networks encompass all these social behaviors that are occurring within a group, they're a really fundamental way to describe complex social interactions. And as I mentioned, the key feature of social networks
when we're thinking from a multi-level selection framework is that they measure both the position that individuals hold within the network and the group structure of all interacting individuals within the network. So every individual holds a position within their social network. So to give an example, here's a network, and the black node in this network represents an individual that's highly connected within its network. So, or if we want to use network language, this individual has high strength. And whereas compared to this gray node, this is going to be representing an individual that has really low strength or is not well connected within its network. And group network structure can also vary. So for example, here's a group with few social connections, or in network terminology has low network density, versus a group that's highly connected or has high network density. And I just gave one example of one way of measuring a social network using strength and network density, but there are many other ways to use social networks to capture aspects of individual and group behaviors. So another way to describe the social connectivity of individuals and groups is with the shortest paths that connect pairs of individuals. So I hope you all can see my cursor. So if we're looking at this network, the shortest path between this gray node and this black node passes through this white node. And so here in individuals betweenness represents the number of shortest paths that pass through an individual. So for example, this black node here has high betweenness as many shortest paths pass through it. Whereas this gray node has a betweenness of 0 as no shortest paths pass through this gray node. And similarly, we can use shortest paths to describe this group network structure. So here, this group has a high average shortest path length, as the shortest paths connecting many individuals in this network are really long. For example, the shortest path between this individual and this individual has to pass through all other individuals in the network. All right, so social networks can be used to describe more than just the connectivity of individuals. They can also capture the cliquishness of individuals and of groups. So here, when we're thinking about local individual cliquishness or local clustering coefficient, this black node is highly cliquish, as many of its social partners are also interacting with each other, whereas this gray node is not very cliquish. None of its social partners are going to be interacting with each other. And then the cliquishness of groups, we can also think about that. So here, that's going to represent the proportion of social partners in a network that also are interacting with one another. So here's a highly cliquish group versus a less cliquish group. And I want to bring your attention to the idea that individual level network traits have analogous group network traits. Okay. Oh no. Sorry, I made a change and we didn't want to do that. Okay. So yeah, so there are these analogous individual and group network traits, and this is really useful when we're applying a multi-level selection approach. And if I just bored you with a lot of network wonkery, apologies, but the main idea here is that social network metrics can describe a lot of different individual and group behaviors, and specifically connectivity and cliquishness behaviors. And social networks not only capture the complexity of social behaviors, but they also have fitness consequences. And so we know from growing literature that both these individual and group level social network traits have fitness consequences. So group level network traits can influence how information or diseases spread within groups, and an individual's position within a network can impact how likely an individual is to contract a disease, to receive information, and also to secure a mate. So in this study, we're quantifying multi-level selection on these analogous individual and group level properties of social networks using contextual analysis. And that's an analysis that uses partial regression to partition selection among these different levels. And we quantified multi-level selection in experimental populations of forked fungus beetles. So forked fungus beetles are sexually dimorphic, and they live on shelf fungus that grow on logs. And beetles consume this fungus tissue. Females oviposit eggs on this fungus surface, larvae develop inside the fungus, and social interactions take place on or near this fungus. So the strong association with wood rotting fungi spatially subdivides wild beetle populations into subpopulations that are centered on these decaying logs, making forked fungus beetles an excellent system to study multi-level selection in. Beetles are also a great system for studying social interactions and social behaviors, social networks, because all of their social interactions are easily observed on these fungus shelves. And we can then use these social interactions, which in this system we defined as any time a beetle is within 5 centimeters of another beetle, to build social networks. And during the past decade, we've observed beetle interactions in the wild and have used these interactions to describe social networks and selection on social networks. And so here is an example of a social network from a wild population that's mapping all the social interactions between males in blue and females in red. And in wild populations, we also see that an individual's position within their network, in this case, how an individual's strength or how many social connections an individual has predicts their mating success. So this data comes from one subpopulation on one log and documents that males with more social interactions or higher strength have higher mating success. But partitioning selection into individual and group components of social networks in wild populations is really challenging, as the population sizes vary widely. And group properties of social networks really closely co-vary with population size. So to disentangle these naturally co-varying influences, we took an experimental approach in order to measure multi-level selection on social network traits. But before I describe this experimental approach, I want to also mention that in wild populations, we see that the strength and direction of selection in males varies across different subpopulations or across these different logs. So in some subpopulations, we see that an individual's males, how connected they are with a network really positively impacts their mating success. And in others, we don't see that same relationship. And one potential driver of this variable selection is how fungus shelves are distributed across the log. So in wild populations, we see that the distribution of fungus varies widely across different logs. And in an experiment that manipulated how the fungus shelves were distributed, the fungus was either distributed in discrete clumps or dispersed more evenly across the log. We found that fungus distribution really dramatically altered beetle space use behavior. So here are two examples of two different beetle home ranges, or this is where a beetle is most likely to be found. And this is from two different beetles from different experimental populations that had these different fungus resource distributions. And to really visualize how this resource distribution affects individual space use behavior, we can then overlay all the beetle home ranges from one experimental population. And we can visually see that the space use behaviors differ between populations that are in environments where the fungus is evenly distributed versus when it's distributed in clumps. And we expect this change in space use behavior to create different selective environments and impact patterns of selection on individual and group behaviors. So specifically, the strength of selection is expected to intensify when resources and beetles are concentrated in space. So in this clumped resource distribution. And so applying this to a multi-level selection framework, we're going to expect that selection at both of these individual and group levels to intensify when fungus resources are clumped due to increased competition for mates in males, or in females, increased competition for egg-laying sites. So in this study, we used social networks of experimental populations of forked fungus beetles to ask two different questions. First, how do individual and group level properties of social networks influence individual fitness? Or in other words, is multi-level selection operating? And then second, does habitat structure influence multi-level selection on these social network properties? And to answer these questions, we created 12 experimental populations of forked fungus beetles. And our populations included an even sex ratio, so 18 females and 18 males, and also had 18 fungus shelves that were farmed in the lab. And as before, to explore how the variation in habitat structure or how these resources are distributed that we observe in the field. Each population received one of two fungus resource distributions. The fungus was either distributed in 3 discrete clumps or more evenly across this artificial log. And then we surveyed these populations 3 times per day for 2 3-week observation periods. And between periods, we switched the fungus resource distribution experienced by each population. And this way we can control for any differences that might exist between these different experimental populations. We then used the social interactions that we observed within each observation period to build social networks. And again, we're defining these social interactions as any time a beetle comes within 5 centimeters of another beetle, and we're not including fitness-related behaviors in our social networks, as these fitness-related behaviors were then used as fitness metrics in our multi-level selection analyses. So the fitness behaviors that we're also observing for male fitness metric was the number of times that we observed a male mate guarding, and this is an indication of a successful copulation. And our female fitness metric was the number of times we observed females laying eggs. And so in total, we observed 7,865 social interactions between males, between females, and also between the sexes. And we find wide variation in both these individual and group network traits that I described before. And so then using the social network metrics and also these metrics of fitness, we performed a contextual analysis to quantify the influence of both individual network position and group network structure on individual fitness in males and in females. And then we additionally quantified how this fungal resource distribution influenced these individual and group level effects. And so this analysis explores how patterns of multi-level selection on social networks vary across both sexes and habitats. And so let's begin by looking at the results in males. So we see a strong relationship between individual strength and male mating success, similar to what I showed us seeing in that one wild population. So more connected males have higher mating success. And mating success has been relativized to mean fitness in these multi-level selection analyses. And we see that this relationship does not vary with the distribution of fungus. So when fungal resources are clumped, indicated here in green, we see the same positive relationship as when the fungal shelves are evenly distributed, indicated in orange. And even though individual strength influence male mating success, we don't see a relationship between the population's network density or how connected the population is and male mating success in either fungal resource distribution treatments. And I just want to make a note about this figure. The size of these points is scaled by the number of individuals that have that value. And we see that the points are arranged in columns because all individuals within a population share the same network density. And we found really similar results when we looked at another measure of social network connectivity, when we look at individual betweenness and average shortest path length. So males with high individual betweenness, they're more connected in their network, they have higher mating success. But different than what we saw with individual strength, this relationship between betweenness and mating success varies with resource distribution. So the strength of selection on individual betweenness is greater when fungus shelves are clumped. And this aligns with our expectation that the strength of selection should intensify when beetles are concentrated around resource clumps and have higher competition for mates. And as before, even though individual betweenness is influencing male mating success, we don't see a relationship between the population's average shortest path length and male mating success in either fungal distribution. And so from these analyses, we can conclude that male connectivity measured as both strength and betweenness within a network predicts male mating success independently from both group and habitat structures. And as a note, we found no selection on local and global clustering coefficient, indicating that cliquishness does not impact male mating success. But we see a really different story emerge when we look at females. So in females, an individual strength within a network has no effect on the number of eggs that a female lays. However, the network density of the population does impact female reproductive success. And this relationship differs across resource distribution treatments. So when fungus is clumped, females in populations with high network density, so these are highly connected populations, lay fewer eggs. So in other words, group level fecundity selection varies across groups, but only in specific habitat structures when these resources are clumped. And we didn't find any selection acting on betweenness or average shortest path length, or on local or global clustering coefficient in females. So we see different patterns of multi-level selection in males and females. So individual level strength contributed to male mating success, but female reproductive success was explained by group level network density. And so by measuring this multi-level selection, we're uncovering a fitness relationship in females that would have been missed by selection analyses that don't consider group traits, that only consider individual traits. And we also see different patterns across habitat structures. So group level selection only operated in females when fungus resources were clumped. And I was a bit puzzled by this result. Specifically, I wondered, why does selection in females occur on the density or the connectedness of a social network, but not on how connected a female is within that network? So one idea is that this relationship may be driven by male-female interactions. So females may lay fewer eggs in networks with higher density because males may act more aggressively in more densely connected networks, which causes females to experience more harassment and lay fewer eggs, regardless of the position that the females hold within the network. And so to test this idea, or to begin to test this idea, we ran the same analysis, which is now here, but in social networks constructed only from female-female social connections. And we found no fitness implications of how connected a female-only network is. And so this suggests that male-female social connections influence the number of eggs that females lay. So males in more densely connected networks may act more aggressively towards females, limiting the number of eggs that the females lay. And consistent with this hypothesis, group level fecundity selection occurred only in environments with clumped resources, where we expect male-male competition for mates and male aggression to be strongest. So to summarize all these results in one place, we find there are different patterns of multi-level selection in males and females. Males with higher strength and higher betweenness within a network, or in other words, more highly connected males, have higher mating success. So individual level selection. But females in more densely connected networks have lower reproductive success. And this only occurs in environments with clumped fungus resources. So we found these opposing patterns of selection operating across levels and between sexes. So this is sexually antagonistic selection across levels, and it reveals the potential for multi-level sexual conflict to shape the evolution of sex-specific social behaviors. So for example, females' dispersal behavior may be to move away from logs with clumped fungus or away from densely connected networks in response to this multi-level sexual conflict. Of course, these patterns of antagonistic multi-level selection on social networks observed in the study also reveals how multi-level selection may shape the evolution of social networks themselves. However, for social networks to respond to multi-level selection, there must be some transmissible component of this network structure across generations. So we need more work to explore the genetic underpinnings of both individual and group social network traits, and whether these traits are genetically correlated, to understand the evolutionary implications of this multi-level selection on social networks that we're observing. But ultimately, these results suggest that this group level selection on group network traits may be an important force that drives the evolution of social behaviors, and it really calls for more empirical studies measuring multi-level selection on social behaviors. And so with that, I'd like to end by acknowledging the several funding sources and also the colleagues I worked closely with on this study, Phoebe Cook, Butch Brody, and Vince Formica. If you want to check out our published manuscript, you can scan this QR code here. And finally, I'd just like to shamelessly plug that I'm recruiting graduate students and postdocs. So I'll be starting a research group at the University of Buffalo this fall. But instead of studying evolutionary processes, my research group will conduct biology education research, primarily focused on evaluating pedagogical approaches aimed at promoting equitable post-secondary biology education. But one research direction that might be of interest to this group is to evaluate how multi-level selection is taught in undergraduate biology courses. So we've really spent a lot of time today thinking about the importance of multi-level selection theory for understanding evolutionary processes, but a question is it included in post-secondary biology courses? And if it's taught, what is being taught? Is it kin selection? Is it group selection, multi-level selection? And is it introduced as a controversial topic? So if you're interested in collaborating on this project or anything else, please feel free to reach out, and also encourage people who are interested in doing grad school or postdoc reaching out as well. And with that, I'm happy to take any questions.
It was a wonderful presentation, Robert. Thank you. Thank you again for that. And I think your future research is also very exciting here. And I know as my time as a pedagogical research, it is a very controversial topic. And I know that they had 5 misconceptions of evolution, that they always taught group selection as one of them. So I know that we definitely need to, create a more nuanced perspective of this in the pedagogical sphere. So thank you again. But I did see one question here, or one hand up. Robert, did you have a question, or did you take that away?
That was just me clapping. Nice talk.
Oh, good. Yes. Yes, definitely warranted.
Go Sabres.
Yes.
Well, I could ask a question. I mean, now so something that comes to mind that you mentioned at the very beginning was the sort of frequency dependence, if you forgive my phrasing it that way, right? So at the very beginning, you pointed out that you're going to be talking about the effect of groups on individual fitness. Was that, do I understand correctly?
Yeah, the effect of both individual and group network metrics on individual fitness.
And that would be opposed to, and here's where I expose my naivete, that would be opposed to a group itself having heritable properties like geographic range, or I mean, Michael Wade was discussing something like that, if I remember correctly in his presentation. But so my question then, Robin, is if do you see evidence for the latter kind of group selection in your study group as opposed to the former?
If I'm understanding your question correctly, so when in these experimental populations, there was not really difference in number of eggs laid or in number of successful matings across these different experimental groups. It was, so in that sense, and like that variance in group level fitness, we're not seeing evidence for that in these experimental populations. In the wild, population size varies widely, and that's something that we controlled for in these experimental populations. And so that's a factor to think on. Yeah, I don't know if I'm addressing your question.
Yeah, no, you are. I mean, I guess I'm very new to this stuff, and please, I'm grateful for everyone's patience to listen to this. But I personally found that really helpful in the Okasha textbook from back in whenever that was. He makes a distinction between MLS1 and MLS2.
Yes.
And from what I've heard so far, the MLS1 seems to, and here's someone will hit me with a hammer, I'm sure, but MLS1 seems to be where we can kind of, it's a bit more intuitive, I guess. Maybe that's the way to phrase it. Whereas MLS2, right, where groups themselves have heritable properties, that, I don't know, that strikes me as a bit harder to really sink my teeth into. So anyways, yes, you answered my question. Thank you very much. But
Robert, just on that topic there, we'll be discussing this next Wednesday. So the distinction between MLS1 and MLS2. And going through a lot of that history, because that's very interesting perspective, because actually MLS2 has been traditionally the more accepted version of multi-level selection, just because it framed it in longer timescales. So this is yeah, we're definitely go through this history in the next Wednesday meeting. So please join us at the initiative here. So but yeah, without further ado, David, are there any other questions or comments?
I'd like to say I can't help but think of Omar Eldakar's work on sexual conflict and water striders. And it's kind of the same story in terms of sexual harassment, basically being the selfish trait in males being sexual harassment. But the study is different. It did not involve social network analysis per se. And it ended up being more of a straightforward MLS1 story. Basically, within each group, the aggressive males did better than the passive males. but groups with passive males were much more productive than groups with aggressive males. So it was a pretty well standard altruism selfishness story. And I have to confess a kind of a ambivalence towards social network analysis, because it doesn't directly measure. I mean, you can measure it and you can get your numbers, but I find it difficult to translate the social network parameters to anything that resembles a natural history story in terms of the actual social behaviors, what's good for the group, what's good for the individual. Do you have that problem too? And I'm happy to have you here to address that question.
Yeah, I guess so to your first part about the Eldekar paper, that was definitely an inspiration for the study. I think, yeah, such a cool paper. And I think something that differs this study from that is the Eldekar paper found, yeah, it's more classic altruism selfishness story, but because it's all, you see these different levels of selection occurring within males. Whereas in this study, we see individual level selection on males and group level selection on females. So that's kind of a distinction between how things are operating. Also in the Eldekar paper, females could disperse to different populations, and that's kind of what created that variation.
Yep.
So yeah, but yeah, definitely a lot of similarities and things to compare those two studies. But yeah, in terms of social networks, I totally hear your frustration and sometimes feel that too, where it's you can measure something about social behaviors, but what is it really measuring? It's a little bit abstracted, but I still think social behaviors are so complex that it's nice to be able to use a network to try to capture that complexity. So it's both. It allows you to capture that complexity, but then because it's doing so, it becomes a little bit more abstracted. But I guess the more that you spend time with social networks, the less abstract it feels. But
Okay. Well, I guess I guess I'm up next. Is that right, Mitch?
Yes, please. You're up next.
Okay. Okay, I could say and now for something completely different, but that's the point. It's not completely different. So let's begin squarely in biology with major evolutionary transitions, something that's been briefly covered. So this is the idea that the balance between levels of selection is not fixed, but can itself evolve. And when these mechanisms evolve to suppress disruptive selection within groups, then between group selection becomes the primary evolutionary force, and groups become so cooperative that they qualify as organisms in their own right. And so this was originally proposed by Lynn Margulis to explain the evolution of eukaryotic cells, and then it was generalized by John Maynard Smith, who previously was a critic of group selection and remained so actually, but not in this case. He saw this as clearly as an example of multi-level selection. So the generalized version explains everything from the origin of life to eusocial insect colonies. And it's against that background that we can segue directly to the human case by thinking of human evolution as a major evolutionary transition. And wonderful work by these authors, among others, shows that even though we share most of our genes with chimpanzees, there's a night and day difference in the degree of cooperation. Physical violence over 100 times more frequent in a chimpanzee community than a hunter-gatherer society. Bonobos are somewhat more peaceable, but still lots of conflict. Why the human case? One probably was a greater need to cooperate in the savanna environment, but mostly it was a matter of social control, that in a small-scale human society, such as almost all hunter-gatherer societies, bullying and other forms of disruptive self-serving behaviors are simply not tolerated. Our species are extremely talented at suppressing disruptive self-serving behavior. And this is increasingly being studied as a form of self-domestication, in just the same way that we bred docile traits in our companion animals. We bred them in our cells. And right away, this framework explains something which is distinctively human, which is the whole concept of morality and moral systems. And what's interesting about morality is that it has two dimensions. There's a coercive dimension, basically that which we're expected to do when we're punished if we don't. And then there's a voluntary dimension based on positive emotions of love, sympathy, so on and so forth. Also, morality is largely a within-group affair. There's a separate set of rules for conduct between groups, which is not always hostile, but often is. And so this whole profile is exactly what's expected from a multi-level selection perspective. And it's so interesting, something I've been wanting to say and for some of these other more strictly biological talks, that the reason that these two dimensions go together is that the coercive dimension makes it safe to practice the voluntary dimension. In the absence of the commercial dimension, it simply would not be safe to behave prosocially towards others. But thanks to the coercive dimension, then you can freely express your other-oriented nature and you will not be hurt. You're less likely to be hurt. And so the entire package that we associate with human moral systems can be understood as part of this major evolutionary transition. And so nearly everything distinctive about our species we can now see as a form of cooperation. Of course, we have physical forms. Mental, when we think about mental traits, so often in psychology, such things as perception, memory, and decision-making are treated as individual level traits, but all of them take place in a social context, especially in hunter-gatherer society, every single one of them. And then especially the capacity for symbolic thought. So one landmark book by Terry Deacon on the symbolic species is establishes that there's so many efforts to identify human uniqueness, what sets us apart from all other species. Most of them fail, but there's something about symbolic thought that actually is very distinctively human, at least in the degree to which we do it. And what it enables is basically an entire separate stream of inheritance. And so this is called dual inheritance theory. And let me walk through it just a little bit slowly. So we all have a genotype, a collection of genes, which influence just about anything you could measure in us, our phenotype. We also have a set of symbols in our head. Let's call it a symbotype to stress the comparison, which interacts with the genotype. That's where such things as epigenetics comes in, and also influences just about everything that can be measured in us, our very same phenotype. And so the symbotype and genotype interact with each other during our lifetimes, and they've been co-evolving with each other for our entire history as a species. And even though human meaning systems, worldviews have been studied in many different disciplines, in the social sciences and humanities, there is tremendous, in my view, metaphorical transfer in thinking of our symbolic systems as the cultural equivalent of our genes. And Terry Deacon has a wonderful phrase. He says that humans are a symbolically eusocial species. In other words, the social glue that holds our groups together is much less genetic relatedness than it is the symbolic systems that bind us together. And the Latin root of religion is religio, which means to bind. So we have this wonderful work that's done, that's taking place. Peter Turchin and others working on the human history as a fossil record of cultural multi-level selection. Wonderful work by Joe Henrich on cultural diversity, the idea that our own Western educated, industrial, rich and democratic societies is very peculiar, but because virtually all of the science and scholarship takes place in weird societies, we've been mistaking our particular cultural form as human nature. And human culture is much more diverse than that. The bushy tree of human culture should be studied in the same way as the bushy tree of biological life. And so there's just so much that's represented in that phrase. And from all of this, in addition to this kind of macro scale of human origins and what it means to be human. There's also tremendous practical applications, which is what I've been devoting myself to for nearly 20 years now. And so here's a couple of my books and then two papers. One is a PNAS paper published last year, and then a paper published this year on economics, rethinking the theoretical foundation of economics. And you'll see the multi-level paradigm is right there in the subtitle. And so when we think about practical applications, here's a slide that I had in my introduction, the simple idea that it motivates multi-level selection in general, the theory in general. And what we can say is that all contemporary groups are faced with the same tensions. That's how fundamental this is, that in any group, any contemporary group, there is a tension between benefiting oneself at the expense of other members of that group, or working for that group to function well in comparison to other groups. And just as social control was needed for the genetic major evolutionary transition, social control is needed in every contemporary group in our life in order to basically suppress the potential for disruptive within group behaviors in order for the group to function as adaptive units. And when you actually study contemporary groups, you find invariably that they vary. There's a bell-shaped curve. There's a high performing end, which is well governed and functions well as a result. There's many groups, some groups, the other end of the bell curve, which just implode. And then most of our groups are muddling around in the middle, working pretty well, but with much room for improvement. And all groups can benefit from learning this overarching perspective. So a key figure in this regard is Elinor Ostrom. She was a political scientist who won the Nobel Prize in Economics in 2009. She was probably the biggest outlier ever to win the Nobel Prize in Economics. What she studied was the tragedy of the commons, in which selfishness, basically cheating, takes the form of taking more than your share of the resource. And received wisdom, economic wisdom, was that the tragedy will always occur unless you can privatize the resource, which you can't always do, or impose top-down regulation. And what Ostrom showed was that actually by studying common pool resource groups, she showed that some, not all, were capable of self-managing their resources, and if they implement certain core design principles. And so her achievement, which won her the Nobel Prize, was to identify these core design principles. And I collaborated with Lynn for 3 years prior to her death. And what we did together was to generalize the core design principles from an evolutionary, multi-level evolutionary perspective. So here they are. I'm going to be very quick in running through them, but just imagine a group that has these properties. They have a clear sense of identity and sense of purpose. They know that they're a group and what the group is about. There's an equitable distribution of benefits and resources, not the case that some get the benefits and others do the work. Decision-making inclusive, not the case that decisions are made that you have no say in. Monitoring of behavior, so we know what we're doing. Graduated responding to helpful and unhelpful behavior. If we're not doing what we should, there's correctives, but those correctives don't start out harsh necessarily. They only escalate as necessary. And also there's abundant rewards for good behavior in addition to mild punishment for bad behavior that escalates as needed. Fast and fair conflict resolutions, which the conflicts will occur. They need to be resolved in a manner that's fair to all parties. And then these 6, or 2 through 6, basically govern social interactions within groups, and then 7 and 8 govern between group relations. First, the group has to have a degree of autonomy, and then there must be relations with other groups which embody the same principles. So I hope you can see what perfect sense this makes from a multi-level perspective. And the fact that these core design principles are scale independent, needed to govern relations between groups in addition to within groups, is a tremendous conceptual simplification. And so this now becomes a kind of a toolkit for helping groups govern themselves better. And let's see how this works in a social environment. Just imagine a school which either does or does not implement these core design principles. Could that make a difference between a terrific school experience or a horrible school experience? And one of my first implementations of these principles was I had an opportunity to help design a school for at-risk youth in my city of Binghamton. This was for 9th and 10th graders who failed at least 3 of their courses during the previous year. That was what you needed to get in. We did a randomized control trial. And importantly, we did not instruct the students in the core design principles. We created a social environment in which we built them in. And so this was an exercise in niche construction. And we didn't have a lot of resources to do this, but we could to a degree. And the results were this tremendous success, basically, that the students in this class, not only did they do far outperform the comparison group in a randomized control trial, but on 3 topics here, algebra, living environment, 4 topics, they performed on a par with the average student in the school district. These were kids that had flunked 3 or more of their classes the previous year. And even more amazing, as you can see from the left graph, this improvement took place during the first quarter. So how is this possible for such a wicked problem that you could not only make a difference, but the difference would take place that fast? And I like to call the answer to this the parable of the turtle. It turns out that all of us, including these at-risk kids, are like sensible turtles, capable of assessing their social environment and pulling into their shells when it's not safe to extend yourself. And so what they did evidently is they basically, they could recognize quickly that the social environment that we provided in the school was a safe and secure social environment that could come out of their shells, and it didn't take them long to do so. And so this is tremendously optimistic conclusion. We've also done a survey study. And in the survey, we asked participants to provide information on 2 groups that they knew well, a workplace group and any other kind of their group of their choice, type of group of their choice. And the information they provided was how well they implement the core design principles, and then how well they perform as a group on these variables of trust, satisfaction, needs, cooperation, and commitment. And the results of this study were a very strong correlation between group performance and implementation of the CDPs. In every study we've done, we find what Ostrom found, that their group performance correlates with the CDPs. Business groups no less than other types of groups, but on average, business groups were deficient in all 8 core design principles, every one, with the biggest deficits being local autonomy, sense of purpose, identity and purpose, and decision-making. So why is it that business groups, which need the principles as well as any other kind of groups, are deficient in all 8? As we'll see, that's due to, I think, a large part to conflicting narratives. And so in addition to our own survey study, it turns out that if you review the literature, then even though Ostrom is little known or cited, it's easy enough to search the literature with keywords indicative of the CBS, of the CDPs. And when you do this, the evidence for the efficacy of the core design principles is simply overwhelming. It's almost as if you don't need to do a single additional study. And this includes well-known business movements such as the B Corp and conscious capitalism movements. And so the fact that businesses on average are deficient in all 8 core design principles, that's just the average. There's still variation around that average. And on the high end of the business curve, you find amazing examples. And just to dwell a little bit on the left-hand book here, Everybody Matters, this is about a manufacturing firm named, called Barry Waymiller. They make stuff, and thanks to Bob Chapman, who's the CEO, It's an exceptionally caring company and functions well as a result. So that's great. But what's totally remarkable about this case is that Barry Waymiller has acquired, but they don't use the word acquire, they use the word adopt. That's how caring they are, how pro-social they are, over 140 other companies, and transform their culture as well. And so here we have, just hiding in plain sight, a natural experiment replicated 140 times of basically pro-social cultural evolution. And unless you're reading business books, you don't know about it. So one of the things we're doing is we're developing a framework for evolutionary case reports, for example, such as this, in which we can understand these in terms of the very general principles. And so one reason why these are hiding, despite the fact that they're in plain sight, is because they're being concealed by conflicting narratives. So I'll end up with just two conflicting narratives. One, of course, is kind of obvious, the neoclassical economic paradigm, which is centered on the concept of the invisible hand. In other words, the idea that the pursuit of lower level interests robustly benefits the common good, as if led by an invisible hand. And so from a multi-level perspective, this is profoundly untrue. It results in demonstrably unproductive practices such as forced distribution ranking. You treat the performance of the individual as if it's an individual level trait. You rank them, you retain the best, you throw out the worst as if they're just like gold nuggets that you're panning for, without any glimmer that performance is based on social interaction. And not only is this the way we conduct our businesses, it's also the way we conduct our education. And so it's shocking when you think of it, that without knowing it, we're setting our children, in addition to us adults, on this treadmill of competition in all the wrong places. And multi-level selection is needed to know that it's not that competition doesn't exist, it just has to be employed in the right places. It's not hard to understand, but you need to have the perspective. Somewhat more surprisingly, there's also a complex system science also has a conflicting narrative, which can be seen by two meanings of the key phrase complex adaptive systems. Meaning one, a complex system that is adaptive as a system. Meaning two, a complex system composed of agents following their respective adaptive strategies. And if you know about multi-level selection theory, you know that in the absence of higher level selection, we're going to get CAS2 systems, that a community of species following their respective adaptive strategies does not add up to a well-functioning community any more than a laissez-faire economy adds up to a well-functioning economy. So that's the critical distinction that multi-level selection makes, is between these two meanings of complex adaptive systems. But do you know there's no clarity on this distinction in the complex adaptive systems literature, where these two meanings are thoroughly intermixed? And with my colleague Guru Madhavan, we demonstrated this by using complex adaptive systems as a search term. We got over 1,000 hits. Then we used complex maladaptive systems as a search term, and we got 0 hits. It's just not a thing. And so this demonstrates the confusion that can be, that comes by not having the multi-level dynamic clearly in mind. So when I talk about this, I almost always include a slide such as this one, which says that multi-level selection is like a perverse alchemist who turns gold into lead, basically. Everything virtuous becomes corrupt and pathological higher up a scale. And so this is one reason why this basic dynamic is so important for people to learn. There's a huge need for basic literacy. And here's one example, a wonderful, wonderful podcast produced by the National Geographic that uses Bill Muir's classic experiments on group selection in hens, and then weaves it in with human examples, both from education and business. So I highly recommend just type in the problem with super chickens. And then here is a peer review article that I wrote in Pro-Social World's online magazine, This View of Life, which actually does, earlier on, a comment in this session was saying, let's not be too dismissive of the critique of group selection by Williams and others. I couldn't agree more. I couldn't agree more. And so in this article, I basically adopt the mantle of G.C. Williams, which might seem ironic, but what I say in the article is that G.C. Williams made two strong claims. The first claim was that higher level adaptation requires a process of higher level selection and should not be invoked otherwise. That claim has withstood the test of time. Everybody needs to know about that claim. The second claim was the empirical claim that higher level selection almost never takes place in the real world. That claim has not withstood the test of time. And so what we need to do is we need to retain the theoretical claim, and then we need to basically decide the empirical claim on a case-by-case basis, as we've seen most recently in Robin's wonderful talk just preceding mine. And it's at that point that multi-level selection will have regained its foundational status. So what I've tried to say in this talk is that evolutionary biologists should be keenly interested in the expansion of Darwinian thinking beyond the biological sciences. It's just an amazing expansion. Multi-level selection is essential for understanding what it means to be human. We're cooperative to the core, and we can only understand that from a multi-level selection perspective. MLS has immense practical utility, and the foundational nature of MLS with respect to human affairs is a strong reason to reestablish its foundational nature within the biological sciences.
Thank you for that, David.
Okay, so
We have our first question here from Vincent.
Vincent.
Hey, David, really interesting stuff. I wondered, after seeing Robin's talk, I was wondering if you had thought about, using a contextual analysis approach where you're kind of partitioning the influence of individual properties on performance and the role that group traits might have on individual performance, right? So in the work of students, like looking at things like study habits or background or whatever, and then looking at the role that the group, the pod, the school, the family might have on performance, say on school exams. Have you thought about partitioning that and seeing which is stronger?
Yeah, well, this is a personal bias. This is a conversation that's taking place in the multi-level selection initiative. So once again, I encourage anyone with an interest. You don't have to be an expert. So please join. But I personally, I'm not fond of any statistical partitioning method, not the price equation, not contextual analysis, because I like to think of multi-level selection as a causal theory. I want to actually identify the traits, which are often highly nonlinear, and how they interact both within and between groups to build causal models. And I think that what Elliott is better at than I am at recounting is that almost by their nature, statistical partitioning methods are going to misclassify traits. They're going to take traits that should not be called group selection, and they make it seem as if they are, like they appear in the group component of the price equation. And I think contextual analysis has its own categorization errors. So I'm not saying that they shouldn't be used, but personally, I just want to know causally what is going on. What are the traits that are evolving? How do they interact with each other? How does that result in fitness differences within groups? And how does it result in differential fitnesses between groups? And in the practical case, I think that this deals with functional organization, studying groups as like organisms, basically, in which case they need a anatomy, physiology, immune system, nervous system, studying groups like organisms. And that is, that kind of functional analysis, I think, is it's not incompatible with statistical partitioning method, but so in some ways I'm just indicating a personal preference.
Thanks.
Yeah, I'm sorry, we're going to have to go to our next presentation here. So MK, if you wouldn't mind asking the question either in the chat or in the Q&A box below, and then we'll get to it. But actually, after Stephen talks here, we will have a few minutes to answer broad questions. So, so if anybody wants to stick around and ask questions, not just about Stevens, which we'll definitely give you 5 minutes or more to, because I know it's very interesting material as well. But after that, to talk about the initiative in broad general terms as well, then please feel free to stick around. But yeah, without further ado, Stephen, if you wouldn't mind.
Okay, let me share my screen here. And there we go. All right. Well, hello, and thanks for having me. This is like a real honor to be able to, be part of this. And, thanks for everybody for putting it together. It's interesting going after David's talk because, you pulled in the human element of it. And I think some of the topics that were discussed there, I'll be traversing broadly. Okay, so here is the title here of the talk. It's a little bit cumbersome, but really the whole idea is to look for dynamics that are multi-level selection type dynamics that historians and sociologists and political scientists are working on without using that term. And I should mention this comes out of the work that we're currently doing. I'm doing with Matteo and AJ as put down there on political ideology. And it does leverage, among other biological theories and other theories in general, MLS to understand, the broad elements of political ideology. So this work is really a joint venture. Okay, so I just wanted to go through why, I'm doing this and why I put this together. The first one is the whole catalysis, consilience, outreach that we've talked about so much in this MLS, initiative. There are people, Steven Pinker and Richard Dawkins, and I picked on Wrangham here, and this is a quote by David. They're, detractors of MLS, and it's very difficult to get them to change opinions or move the needle on them. But I think that there's a whole nother set of people that we can spend time on, which is again historians, political scientists who are writing about the problems and topics of MLS, and they're not really conversant with the theory, so they're writing about the dynamics without having the biological theory. So I think it's a bigger group. I think it's low-hanging fruit. I think they're not recalcitrant. They're not, I don't want to say indoctrinated, but they're not coming at this with that deep skepticism that some evolutionary biologists and other people in the field do come with if they've been, reading about some of these naive group selection topics. Okay, so this really comes out of, it's a little bit, I must say, inspired by one of the chapters we were working on where, trying to talk about MLS theory. I think that's always a hard thing to do, to talk about MLS theory to somebody who maybe isn't conversant with it on any level. And AJ had a comment, one of my co-authors there, for those of you who don't know him, AJ Figueredo, he said, meet people where they're at, start talking about what they know already. And I remember, that is very good advice, and something I used to love when my dissertation advisor would do that for me and kind of label something I already knew and link it to the literature. And this is one example of that kind of outreach. And if you want, you can read down below. This is an email I wrote a couple of months ago after reading this book called The Story of Constitutions. And it's one that I will review. And yeah, I just saw that he was reaching out, this author who was writing, he's a political scientist, he's writing about constitutions, he's talking about Dunbar's number, ostracism, trust, group size, and all these related issues that were really central to MLS. again, without ever mentioning it, not because of any prejudice, but just because it seems that he's never been exposed to it. All right. So here's the second purpose of why I put this together. Mitch talked about this actually, I think, just before, and also a week or so ago, the idea of external validity. You know, and I see this as a two-way street. There's mutual need. We can offer a framework of biological understanding that will help them interpret what they're talking about and debating. And then on the other hand, the history can offer us an empirical laboratory. So that's going to supplement theory, modeling, and the animal studies that we traditionally rely on. And then the third purpose is to sort of jumpstart this historical political science subgroup. And we've been talking about this a lot, and we've been sort of preoccupied, but it is one of the things that we wanted to do. And I don't mean for you to jump in with questions right now, but yeah, anybody at the end who's interested or has other publications, I'm going to go through a whole series of publications. And if you have any additional recommendations, be happy to hear those. Okay, this is sort of a rundown of the topics. I won't go through these, but I loosely group them into these headings, you might say. And then under each heading, I'll just show you a couple of books and then representative quotes from those books to again show that these are authors that are talking about MLS type dynamics that could really benefit from the literature on MLS, and that we could benefit from reading them and getting, you know, some historical nuance and some flavor of their literatures and how they're broaching these very same questions about order and societies and things like strong reciprocity and punishment and prosociality, how we get to an ordered society, and how we do so while preserving some level of individual autonomy and freedom. Okay, so here's the first of them, power and war. Okay, Bertrand de Juvenal. He has this very difficult to read book, but really insightful. If you have the patience to go through it, it's very florid. There's lots of flourishes, but he talks about political society as sort of these social pyramids that interact at the summit, meaning you know, at the core of government. And he talks about the breakup of natural groups and the members, which were divided into categories to be individual capacities, right? Soldiers, taxpayers, and electors, and how power, the power of the state and the king, sort of war with those lower level elements like the clan and the family. And that's been sort of a feature of history. Okay, next one. This is one we talked about in our 2020 multi-level selection book. This quote is from there, actually. Oh, and it's from Charles Tilly. His famous quote is "War made the state, and the state made war," and he discusses aggregating impulses induced by conflict. So it's called the bellocentric warfare paradigm, and it finds the outlines of the state called forth by necessity in the form of military administration, organization, financing, credit, banking, martial law. I mean, this is kind of a Red Queen type effect, right? Competition from one group necessitates your leveling up in terms of societal organization and administration and warfare. All right, next. This is a really great book called Fear of Enemies and Collective Action. It's a long quote, might be well worth going through. He says such accounts consider groups to be the natural unit of social analysis, and therefore regard the political group as the result of an agglomeration of natural groups such as families and clans. The choice between solitary individual and the small group is as irreducible unit of social analysis determines the character and direction of a political theory. And then he goes on to talk about both individualistic and group accounts eventually agree that their respective fundamental unit of analysis will sooner or later become incapable of providing for their own survival and security, And then again, kind of in the direction of Tilly, he's talking about agglomerations for mutual need, why societies then grow, why individuals cohere into larger and larger groups. All right, and next up, here's a different kind of class of books, constitutional order and state formation. So these next books are sort of caught up in this whole idea of creating that constitutional cage, that order, that structure that society is formed around. All right, this is the person I wrote to. He wrote this very nice book, The Story of Constitutions. And he wrote, of the many, many quotes I could have put in here, a higher degree of political organization brings internal and external advantages. A shared belief in abstract concepts such as religion, law, ranks, and classes unites group members and at the same time promotes acceptance of authority and non-egalitarian relations. A group with a higher degree of political organization can produce and accumulate more resources and organize and arm itself much better in many different areas of human competition. It can thus defend itself better, and it can even conquer and exploit other groups. Here's an older book by McNeill. I'm a fan of his work. He's got many, many interesting books. This one I picked out called Polyethnicity and the Natural Unity in World History. And he talks about sort of that myth of national brotherhood and ethnic unity, promoting self-sacrifice in war and sustained peace at home. Okay, and then there's Hegel. I've just been finally reading original Hegel. I find it very difficult, as most people do, I think. So The Philosophy of Right, the sort of book that he most pointedly talked about this whole idea of the state being an organic whole. And he talks about the nation as having, he doesn't, I don't think use the word, but emergent properties. And he, in general, sort of shades towards this idea that, yeah, the state is larger than the sum of its parts, and that people get together and they become sort of something of a superorganism. Okay, next up is this book by Elkins, Ginsburg, and Melton called The Endurance of National Constitutions. So briefly, they say, again, among other great quotes, constitutions are meant to hold together disparate groups with different needs, preferences, and goals. And in that book, they're talking about ethnic diversity, cultural diversity, size, and population, and they're discussing how constitutions help keep especially pluralistic democratic societies together. Okay, now there's a whole series, there's a lot of these books on nationalism, solidarity, and the creation of state-level cohesion. Sorry if I'm running through these fast, there's just a lot of them. Okay, so one of them is this book by Benedict Anderson, which has a lot of fame within his own circle. And he's really talking about the creation of sort of statehood from an imagination, almost like a mythic standpoint. So he says that the new synchronic novelty could arise historically only when substantial groups of people were in position to think of themselves as living parallel lives to those other substantial groups of people. Then he goes on to talk about all these technological innovations like communication and navigation and maps and ships and all of these systems that link people up. And he's saying this is why nationalism became something that we talked about and saw, you know, in modern societies, because we had these capabilities. Now, he is sort of part, if you look to the left here, hopefully you can see my cursor, you'll see 5 different authors of which he's a part. And all of these authors talk about nationalism as being something that's created. They talk about it being sort of a product of late 18th and 19th century origins, linking it to sort of the French Revolution. They emphasize myth, state power, printing, travel, communication as being able to weld lower level units together. And then here, this slide actually talks about Hastings, Adrian Hastings, who pushes back against that, and, you know, while acknowledging all of it, talks about nationalism as predating that, at least the seeds of nationalism coming from an earlier point in time. And what he says, it arises chiefly where and when a particular ethnicity or nation feels itself threatened in regard to its own proper character, extent, or importance. Okay, moving on. Okay, so here is another person, I think, Caspar Hershey, The Origins of Nationalism, who, a lot like Hastings, is trying to kind of mediate between these different groups, between, you know, Benedict Anderson and those people that would have nationalism as simply a mythic invention of modernity, and others that look at something that you might call perennial or primordial nationalism, that looks at ethnicity and long history as the origins of nationalism. So he's trying to take a balanced view between those two different camps. Okay, and then there's Anthony D. Smith, really prolific author on nationalism, and he says, is here conceived of as a political discourse constructed by chronically failing would-be empires stuck in a battle to keep each other at bay. And there's a later book in here by Wimmer, and he talks about the same thing, as these empires, you know, fighting for territory and warring and actually creating nationalism through that process as people try to keep stability over their own territory. As these empires try to co-opt power, they become kind of nationalistic, and they gain solidarity and coherence by pushing back against that larger group. Okay, here was an interesting book I just came across not too long ago, by Matthew Rose, and he is sort of a liberal libertarian type person, taking a look at these 5 different philosophers of the radical right, trying to see, well, you know, is there anything that we should be engaging with, anything of value in them? And he talks about a wide array of them. They really have very little coherence, except for them being on the far right, but Spangler, Evola, Yaki, Benoist, and Samuel Francis. And he basically looks at them, and I think one of the commonalities that you would see if you read this book or some of the original authors is that they have an emphasis on solidarity. They're often all talking about solidarity, at least among a subgroup of the population, and how you know, there needs to be unity, etc. And they're deploring sometimes you know, the pluralism in society. And so Rose writes, you know, political life does not depend on truths or values that transcend our identities, the radical right claimed, right? He's representing their views. And instead, it depends on recognizing that human identity at its most primordial level is something inherited. And again, that's his representation of what is common to all 5 of those authors. Okay, moving on. Here we have a book called The General Will. And that might remind you of like Rousseau, the General Will. And this is by Farr and Williams. And they say, if the general will is only general or universal with respect to some particular political community, then it will inevitably be partial with respect to subgroups within a nation, not to mention towards members of other political communities. So then he has this follow-up quote, which I think is important. He talks about Scheller and sympathy towards members of the in-group, but the flip side of that being resentment towards outsiders. And then he kind of tries to contrast what can be hard to differentiate because of that dynamic: patriotism versus nationalism, and the in-group solidarity that you feel, and its natural kind of corollary, which could be out-group differentiation and possibly vilification. Okay, here is a book by Simmel, and it's called Conflict. And he really is talking, you'll see the quote there, the group in a state of peace can permit antagonistic members to live with one another in an undecided situation because each of them can go his own way and can avoid collisions. But then a state of conflict, however, pulls members so tightly together and subjects them to such a uniform impulse that they either must completely get along with or completely repel one another. So he's really talking about the differing demands of war and peace, and how external conflicts will bring a lower level of internal tolerance for group differences, for plurality, and so on. Okay, now moving on here to empires and nations. Here's that book by Wimmer that I just mentioned, called Waves of War. And this is trying to, he explains this in his introduction right here. He's seeking to explain the transformation of the political world from a world of multi-ethnic empires, dynastic kingdoms, tribal confederacies, and city-states to a world of states each ruled in the name of a nation properly seated in a General Assembly of the UN. So, you know, the whole rise effectively of the nation state and how we went from these lower level groupings to large empires and then back down to this lower level of state unity, or the nation state as a level once removed from empires. Okay, and here we have this book called Becoming International by Bartleson. What he's basically doing is he's contrasting the Enlightenment rationalist cosmopolitan view that you're a citizen of the world with the Romantic movement and their sort of emotional particularism that talked about more of a distinct organic history that people had. So it's well worth, I mean, that's the main theme of the book, but it's brought out historically and philosophically in a really nice way. And then there's Adam Smith and Wealth of Nations, really talking about individuals becoming more specialized atoms in an ever increasing productive whole, right? So instead of being these more autonomous units that can farm and fish and take care of their families and deal with external threats, you get these productive societies that increasingly, you know, find the individual and place them into more of a production line. And, you know, just like the pin making example in Smith, you know, they gain more productivity, but on the other hand, they lose generalizability and autonomy. Okay, running out of time, but we're getting there. So collective action problems in states. This is a book called The Problem of Social Order. And there's these 6 sort of ideas about how we gain social order that, you know, these are basically what Wrong is saying. And I'm paraphrasing here, but you have coercion by force or by propaganda. You have natural cohesion by shared genetics or by shared culture and tradition. Or number 5, you have mutual advantage and economic interest. Or in number 6, you have dynamic tension, where there's competing interests checking one another. This is a really interesting book. You know, you pick up these books and you flip through and you look in the index for MLS and it's not there, and you read the whole thing and you just can't believe it was written without MLS theory. Hector and Rhodes, Theories of Social Order. They're talking about all these wonderful theorists, Weber and Durkheim and Tocqueville and Gellner, etc. And they write, the challenge is that behaving cooperatively may impose costs on the individual, as we know in MLS theory, right? So they ask that main question that we've all asked: how can societies promote high levels of coordination and cooperation? Okay, and by the way, they get as far as citing Gintis, Axelrod, Hayek, but again, they didn't get right there. Here is Simmel again, because this book is actually 2 books in one. He wrote this shorter book called The Web of Group Affiliations, which is really just as relevant. He says, an advanced culture broadens more and more the social groups to which we belong with our whole personality. But at the same time, the individual is deprived of many supporters and advantages associated with the more tightly knit primary group. So he's basically talking about a trade-off between depth and breadth of affiliation, from when you move from more small integrated tribal communities to larger social agglomerations like the nation states that we currently live in. Okay, and last set, we have the sovereign state and its competitors. he's talking about lower levels of biological aggregation, and then he's talking about things that we would recognize as Red Queen effects and ultrasociality, and that whole evolutionary ratcheting up of social organization, again, not using the terms. This is Spencer, many, many years ago. Societies agree with individual organisms in 4 conspicuous peculiarities. We have size, complexity, interdependence, and institutional longevity. And here's another great example, nations without states. Contemporary politics are grounded upon a constant tension between the pressures exerted by sub-state movements, which put forward nationalistic claims, thus questioning the state's legitimacy, and the growth of supranational organizations, which require the state to surrender relevant attributes of its traditional sovereignty. So what you're having here is the nation state being pulled downwards by these sub-state national type movements, and then upwards in the ladder of aggregation to globalist type institutions. This is a great book talking about the pluralists, which are a whole group of people, and David Runciman talks about 5 of them, and he's discussing the group unit or group person must contain other units or persons, including the person of other groups. So pluralism is effectively a whole philosophy that really reoriented the conversation away from nation states and towards individuals and lower level groups, which we would all agree, I'm sure, have some power and autonomy, and they interact with the state. And then this is one of those 5. This is Harold Lasky, one of those 5 pluralists that Runciman talks about. And he has this book called Studies in the Problem of Sovereignty. And he says, yet, so we are told the state itself, the society of which they form part, is mysteriously one above them. And what he's saying, is that, well, maybe the state isn't this strange Hegelian agentic whole. And what he then talks about is this kind of nested level, that Russian doll level of identity. We represent a state as vast series of concentric circles, each one enveloping the other as we move from individual to family, from family to village, from village to city to county, and the all-embracing state. So I am just about done. Let me just sort of end by talking about some of the more leisurely, or what you'll see is some of these main themes come up over and over again. So pluralism, having individuals in lower level groups with no meta properties assigned to the larger whole or the state, versus other theorists that talk about these reified states that kind of look at individuals welded into a superorganism. You have themes like anarchy versus tyranny, group size versus group cohesion, people talking about individual freedoms versus state survival. And then also these authors that sometimes see people as pluripotent atoms versus specialized cells. So really, the question is, what can an MLS framework offer? And then also, I think we're going to be talking next week about MLS 1 and 2. And I think that's a really relevant debate to this, which we should keep in mind. So I have done, and thank you so much for listening.
Man, that's a great final talk for this wonderful symposium. I'm so proud to have captured this, not only for ourselves, but for recording. Hermina, you've had your hand raised a long time.
I have to unmute, yes. I had a question for the speaker. I just noticed that there was like a lot of similar themes in the books that you chose. And it was also, this got me, I started thinking about it because I was thinking of political anarchism, which for me, and MLS seem very similar to each other. And then it's funny to see it on your last slide. But so that's why it got me started thinking about it. But then another question that I had was but also the themes are similar. And is that just because of your interests, or is that the only place where you think that that multi-level selection can contribute? Or I don't know, I just wanted to understand.
Yeah, no, it's just more of an artifact of what I've been reading over the last, year or so. We had written a 2020 book that didn't really discuss many of these themes at all. And now we're writing another book that does kind of involve them. And yeah, they were just sort of a collection of authors with, really meant to represent the general, overview of what's out there. And broadly just present it to just show that, well, these various people, and they have different opinions, they're from this side or that side, and they have extremely different beliefs, but they're, all talking about what we're talking about. And I think, we would be of some value in that conversation. And I think that their conversations would be of value to us and what they're saying and talking about in the historical examples they cite.
Okay.
Yeah.
Thanks.
Rich?
Yeah, no, I love this research. And I think it's, it's what's it called in psychology, like the Bader-Meinhof phenomenon. Like once you learn of a word, then you begin seeing it everywhere. I think that's the term. Maybe I'm doing it a grave injustice here, but it was also after reading This View of Life, and then reading a few articles by David, and then also talking to you, Stephen, I began seeing multi-level selective dynamics just everywhere. I mean, explaining the tensions in the United States right now, why we don't have one broad social identity, and but also, yeah, what we were talking about with Adam Smith, just the fundaments of modern society are based on a multi-level selective prediction that the benefit of the baker, the butcher, and whatever the end of the quote is in Wealth of Nations, that will benefit the common good, is basically the argument that he's making. So I think it's a multi-level selective argument, but really from an individual level perspective. So I think I love this research that you can just find this everywhere, and I think it's the same to the genetic or the history of biology as well. You see multi-level selection thinking in all these previous thinkers, but they weren't explicitly talking about it as multi-level selection theory. So like, as they say in psychology, it has a long past, but just a very short history. So I think, I love this. So David, if you want to go again.
It just validates its importance, I would add, that this is what we're talking about through MLS is really important to other people, and it's a perennial question.
I think that I'm really sensitive to a comment that was made earlier today about that we don't want to be like scolding or dissing the history of evolutionary biology, and I'm sympathetic to that to a degree, but I'm also feel that without being mean-spirited about it, that some aspects of the history of evolutionary biology are really problematic. And we can begin with the modern synthesis, which has also been called the Great Constriction. So yay, modern synthesis, but with the advent of genetics, the entire study of evolution became gene-centric. And so this kind of inquiry with human cultures and everything that you've just reviewed, it did not take place within evolutionary biology. It took place in other disciplines, and that's where it stands. And so now it's only within the last, since the 1970s, that's what I call generalized Darwinism, that all of a sudden the study of human cultural evolution is a thing from a formal evolutionary perspective. So 50 years were lost, okay? And I'm not blaming anyone, but I'm just noting that 50 years were lost. And so we're playing catch up in a sense, and we're dealing with other disciplines, many other disciplines that have been covering this material, and they can't avoid multi-level selection because that's the kind of species we are.
Exactly.
And so they're converging upon it again and again and again. And I guess my question for you would be, so since they are smart people and they're close to the material, what's the added value of a formal evolutionary perspective? They do seem to be saying the same thing in their own words. And so what, in your view, Stephen, can an evolutionist contribute to this conversation, which is already in some ways well covered by scholars of all these other disciplines.
Yeah, I mean, I think we would have to be humble and also, you know, tentative about what we're offering. I mean, I think we can learn probably more from this literature than we can provide to it. That's the first thing. You know, I think the historical laboratory idea is just so important. But I think that's one of the kind of questions sort of I had, exactly that, you know, like, well, how do you approach this and what can we offer and what can we add? And I think it's generally that sort of unifying biological framework and that common language, because all of these people, while they're talking about the same thing, they're having very disparate kind of arguments, and the literature sets that I'm bringing together are not necessarily even in conversation with each other all the time. And they're certainly creating their own languages, you know, just like Wrangham did, using the word groupishness. They're creating their own languages. So I think we could provide sort of a biological grounding and a common vocabulary. And then, you know, on the other end, we can gain much from them. But I think it's something to talk about more in general, to see, yeah, what can we offer and how do we broach that?
Well, to bring it full circle, I think, and this is one of the things that excites me a lot, is that we could actually be thinking of this enterprise as a form of cultural multi-level selection. That basically scientific inquiry is a form of cultural multi-level selection. And what we have is this huge metapopulation of disciplines and schools of thought within disciplines. And just like Mark Wade's experiment, each configuration of ideas, each formulation is different, complex interactions using the same words in different ways, different historical origins, and that some unification. So therefore a Tower of Babel. So I use the archipelago metaphor all the time in terms of many islands of thought with little communication among islands. And that's the current situation in which we need to perform some kind of higher level selection of unifying these ideas and so on. And unless it's done right, it will take decades or will not take place at all. And I think when you look at the pace of scientific change, what you find is decadal and doesn't have to be that way necessarily. And so I think the idea of understanding this well enough, science well enough, from a cultural multi-level perspective will enable us to accelerate it. So, and I think that's the hope I have for what we're doing, including the multi-level initiative, is we actually make this go faster. We already have. And this symposium is an example. So I'm going to let that be my final words. Anyone else want to, Mitch, you should speak, and anyone else here, including our speakers, should make a final reflection. Anyone who wants to make a final reflection.
Well, no, I guess the one thing that just comes to mind here is that when you're talking about when you call the modern synthesis the great constriction, I know a lot of recent philosophical work on the notion of scientific process sees whatever you view as a paradigm or a scientific revolution or a paradigm shift or any sort of progress in science really comes from the convergence of different causal perspectives. And I think like this is just a testament to that, this whole meeting that we're having right now, because it's just so many different causal perspectives that are coming together. And I mean, everything that you're talking about, Stephen, too, works so well with what David was talking about in the core design principles that lie at the base of all these books, even though they weren't explicitly mentioning it too. So yeah, no, just the convergence between all these different causal perspectives, I really love. So but with saying that, I mean, yes, we will put a link here about more information on the initiative. It is mostly at Prosocial World, but Prosocial World is an online endeavor. So
we're the hub. Prosocial World is the hub, but we intend to be exactly that. So in social network terms.
Good. No, and so if anybody, Michael, César, if you guys want to say a little bit, a little, closing statement, but I also want to open this up to everybody to ask just any questions on multi-level selection, on the initiative, on anything that you might have. So, thank you, César, performing my job much better than I can. So, yeah, this anybody with questions, or Michael, César, if you want to jump in and have some closing comments, feel free to. no closing questions?
Okay, it's been a long day, so maybe we should just bid people farewell and then encourage you to join the multi-level initiative where the conversation will be ongoing, continuous improvement.
Excellent.
Yes, it will. So thank you everybody for coming, and yeah, we'll stop there, but also feel free to ask us more questions at the email and that URL as well. So thank you very much.
We can
Excellent.
stop the
Thank you.
recording now, or yeah, I guess I
Ye-
can do that. Yeah, you can do
Yep.
that, Mike. That's-
Thank